Well. Welcome back, everybody, to day 8 of biochemistry, and hopefully you're all learning a lot. And remember, we don't memorize here. We don't do mnemonic type stuff here because we keep those to a minimum, and we're here to learn and to understand and to get to that mystery word called why things happen. So that is what we're here for in these 8 days so far. And now where we're at is we spent a lot of time getting into molecular biology, about talking about genes and RNA and DNA and that kind of stuff. And we spent several days talking about genetics and all those chromosomal abnormalities. And then we got into the issue of enzymes and hormones in chapters 8 and 9. And then yesterday we finished off talking about vitamins and so on. And so again, all that stuff is setting the stage for metabolism, where we entered metabolism yesterday. So we started talking about metabolism, and specifically we got into carbohydrate metabolism. So that's what we're going to be doing in this first couple hours, is more carbohydrate pathways. And you want to be around for these because there's a lot of pathology coming up in these carbohydrate pathways coming up. And then after that, later on this evening, we'll get into fatty acid and triglyceride metabolism, and we'll start the cholesterol metabolism. And you want to be there for that too. I mean, you don't want to go around. You want to hang around over here because I'm going to make it worth your while. Okay, so the idea is, because you all trust me, you all said you trusted me, you know I got your back, you know I'm going to tell you what you need to know and what you don't need to know. And speaking of that kind of stuff, right here on chapter 13 is a pathway. The citric acid cycle, also called the Krebs cycle, also called the tricarboxylic acid cycle, TCA cycle, whatever you want to call it. Different strokes for different folks, whatever you want to call it. But the important feature is of the Krebs cycle is it's one of the two most important pathways of human life. The first one we talked about was glycolysis yesterday, and now we're going to get into the Krebs cycle, which is even more important than the glycolysis. For life, it is the single most important pathway of them all. Now, I know what you're probably thinking. Oh, no, the Krebs cycle, the citric acid cycle. We heard that so many times in my career, and we never talk about it in pathology or pharmacology. You never hear about the Krebs cycle in any other discipline here. And you think, oh, this is going to be painful. Now, let me say something about the citric acid cycle. You know why you'll never hear about it in pathology or pharmacology or any other discipline? Because it is the single most important pathway of them all. Any biochemical defect, even a partial enzyme defect, is incompatible with life. Yes, there can be a gene defect, but the fetus is miscarried. It's incompatible with life. You will never see the cycle in pathology from Dr. Barone because it's incompatible with life. It is the single most important pathway of them all. So what I'm getting at is, and I'll explain why it's important in just a minute here. But the idea here is even though we talk about the citric acid cycle in biochemistry and you don't in other disciplines, it doesn't mean it's not important. It is important for life. So give it the respect it deserves. And in fact, someday in your lifetime, someday in your life, when you're walking down your clinic hallway with one of your colleagues, and then out of the clear blue sky, do me a favor, out of the clear blue sky, ask your colleague friend, hey, what do you think about the Krebs cycle? What do you think about the TCA cycle? I guarantee you, most likely your buddy friend was going to say, oh, the Krebs cycle, the citric acid cycle, who cares? That's something they talk about in biochemistry. Who cares about it? Do me a favor and tell your buddy friend, you're ignorant. and do it in a nice way, obviously. But they're ignorant. They don't understand how important this pathway is, the citric acid cycle. But the important feature coming up here, because it is the single most important pathway of them all, and I'll tell you why in just a minute here. But the idea is now you're probably thinking right now, oh no, we're going to go through all the cycles and all the enzymes, and you're probably starting to cry right now. You're probably being a lot like this guy. You're being a lot like this guy right here. You know, you're probably starting to cry. Oh no, we got to go through the citric acid cycle. Don't worry, don't be like this guy over here. Don't be a whiner like this guy over here. You know why you don't have to whine about it? Because we're not going to cover it. Because so don't worry about it. So don't be like this guy. I got your back. Don't be like this guy. So we're not going to cover it because you're not going to see it on board exams. Because there's no integration with other disciplines. Because there's no pathology associated with it. You're dead if you don't have the cycle. All right, now what is this? Now what does the citric acid cycle do, and why is it so important? And it's right here in a nutshell. So even though we're going to skip the pathway, it's in your book. You can look at it if you're a type A personality. You've got to know everything. You can look at it in your book. But this is why it's essential for life. In it is where you take the 2 carbons of the acetate of acetyl-CoA. They are coming from carbohydrates, fatty acids, and amino acids. They generate acetyl-CoA. And then in the cycle, there are a series of enzymes that systematically and in an orderly fashion pluck out the high energy electrons of both carbons of the acetate. You'll blow off both carbons as dead end CO2. And the important feature is you put those high energy electrons into NAD to form NADH and FAD to form FADH2. And then they go through the electron transport chain. And then they generate a total of 12 ATPs equivalents per cycle. This is why, in a nutshell, why the citric acid cycle is essential for life. Because again, as we talked about yesterday, your body every second needs trillions and trillions of ATP to do muscle contraction, biosynthetic work, and active transport systems. Trillions and trillions of ATP. And most of them, by far, come from the citric acid cycle. So if there's and that's why it's essential for life, right here. If there's something wrong with the citric acid cycle, you cannot generate enough ATP, and therefore the body cannot survive. This is why the citric acid cycle is essential for life. But for the boards, not that important because there's no pathology. All right, now where we're going though, so we're going to skip over the cycle. I'm making life simple for you. But what we're going to do here though is we're going to I am scared of biochemistry and therefore is boring, but Dr. T is nothing but spicy. Okay. I was reading what that was. It's not boring. It's not boring. Biochemistry is exciting. Make it your friend. It explains pathology. It explains all that pharmacology as well and physiology if you understand biochemistry. Now, where we're going with this is, so even though we're going to skip over the citric acid cycle, we're going to get into oxidative phosphorylation because there's a lot of integration with physiology and pathology and pharmacology. And so the idea is when high energy electrons are being plucked out of carbon and put onto the mobile carriers called NADH and FADH2, the idea now becomes how do you then do the electron transport chain, separate the energy from the electron, and then the electron goes on to the oxygen, and the energy is captured to make ATP. How does that happen? The biochemistry of that. Now that's coming up on the next figure, the first figure tonight. And don't go panicking. When you see the figure, I'll tell you what you need to know. All right, so here is the figure. Don't go panicking. This is the figure of oxidative phosphorylation. Now to orient ourselves, here is the inner mitochondrial membrane where the electron transport chain is located, and on your left side is the mitochondrial matrix, and in the matrix are all these pathways, and where you where like the Krebs cycle, the amino acid oxidation, fatty acid oxidation occurs in the mitochondrial matrix, where high energy electrons are being plucked out of carbon. And on your right side is the cytoplasmic side. All right, now let me do the biochemistry first. And look at the right side. Look at all the things listed here that affect oxidative phosphorylation. Major stuff for board exams. Okay, now what happens there is in the matrix, there are enzymes that are plucking out high energy electrons of the carbon of the food that you eat. Those high energy electrons, for the most part, are put onto NAD to form NADH. NADH then will leave those enzymes, swim over here to the inner mitochondrial membrane, and deposit the high energy electrons into complex 1. Now, because it's a complex, that means there are a number of components, but for the boards, they're not important. Don't worry about the components of complex 1. All right, now then what happens is those electrons, those high energy electrons in complex 1 are then transferred to coenzyme Q, also called ubiquinone, also called CoQ, also called CoQ10. Different names for the same thing. Coenzyme Q, ubiquinone, all the same thing. Now before we move on, other high energy electrons can be given to CoQ, and these are sometimes called the bypass enzymes. And they all generate FADH2, which bypass complex 1 and gives electrons directly to CoQ. One of them is a enzyme, here it's called complex 2. That enzyme is also part of the Krebs cycle, which we purposely skipped. Low yield, that generates FADH2, which gives electrons to CoQ. As we'll talk about tomorrow, there is a major enzyme in beta oxidation of fatty acids. Major pathology tomorrow on this enzyme. Don't worry about it now though. But that enzyme in beta oxidation also generates FADH2, which gives electrons to CoQ. And then there's a shuttle system that's an accessory to glycolysis, also low yield, that generates FADH2 and bypasses complex 1 and gives electrons to ubiquinone by bypassing complex 1. But from here on, everything is the same. So then there's electrons in ubiquinone are then given to complex 3. Now again, because it's a complex, there are a number of components. Don't worry about the individual components in complex 3. And then there's electrons are transferred to cytochrome C. From there, they're transferred to complex 4. In complex 4, again, a number of components. You see some of them listed here, like cytochrome oxidase, cytochrome AA3, which has copper in there. They're not important, with one notable exception. You must know in complex 4, one of the cytochromes in it has iron in it. And that iron, I'm going to show the iron here, is iron in the +3 state, ferric iron. But the important feature here is that when electrons come into complex 4, the electrons are flowing, they're flowing within complex 4. They're going from one component to another component within complex 4. And one of the components is a cytochrome that has iron in the +3 state. When the electron comes into that iron, the iron valency goes down to +2, ferrous iron. And then the electron is then transferred to the oxygen that you breathe in, and the valency of iron goes back to +3. Another electron comes in, the valency goes down to +2. Gives the electron to the oxygen, the valency goes back to +3. Another electron comes in, the valency goes down to +2, gives the electron to the oxygen, the valency goes back to +3. So the valency of that iron is reciprocating. It's going from +3 to +2 to +3 to +2 to +3, +2, +3, +2, +3 as electrons flow through it on the way to the oxygen. Now, if you're thinking, who cares? Who cares? You got to know, because you need to know that, because that's how cyanide kills. And in a few minutes, we're going to talk about cyanide killing, and then I'm going to ask about all the pharmacology that you can use to treat cyanide poisoning. And to understand the to understand the killing of cyanide, you must understand the valency of iron is reciprocating in complex form. But right now, we're just doing the biochemistry. Don't worry about the pharmacology yet, but we will before the end of the page. All right, now at the top of the figure here to the middle of the figure, what you see here is electron transport. How electrons are flowing from NADH to complex 1, to coenzyme Q, to complex 3, to cytochrome C, to complex 4, all the way to the oxygen. That's electricity. That's electron transport. That's electricity. And by the time those electrons go on to the oxygen, they are low energy electrons. Initially, they were high energy electrons in the NADH, which comes from the carbon of the food that you eat. By the time those electrons go on to oxygen, they are low energy electrons. Then you got to ask, well, what happened to the energy? Well, what happens to the energy is the energy as electrons are flowing, just like electricity, the energy is being used to do something. And what the energy is being used and to pump protons from the matrix to the outside. So as electrons are flowing, the energy is then transferred from the electron and being transferred to pumping protons from the matrix to the outside. And now on the outside, that's where the energy resides. It's in the proton gradient. Any gradient has energy. Because there's a lot more protons on the outside than the inside of the matrix, that's where the energy resides, in the proton gradient. Initially, it was associated with an electron of the carbon of the food that you ate. But now the energy is in the proton gradient. All right, now like any gradient, the protons on the outside with their energy want to go back into the matrix to balance out the equilibrium. But they can't because of this thing called the inner mitochondrial membrane. The only way they can go back into the matrix is the protons can go through a little channel called the F0 channel. The channel can open up. The protons can now go through the channel, through the F0 channel, and then they come into a little bleb, little compartment called the F1 compartment. In histology, this is sometimes called the elementary body. It's on the inner side of the inner mitochondrial membrane, like a little bleb, and in that little bleb, little chamber, there is an enzyme that now captures roughly half the energy of the proton and uses it to phosphorylate ADP to form ATP. So half the energy is captured, and the other half the energy is then given off waste that you capture to make ATP, and the other half the energy is given off to maintain your body temperature at 98.6. All right, now these two words, oxidative phosphorylation, what do those words mean? And so it's the oxidative means electron transport. That's the oxidative part, and it's coupled with the phosphorylation of, down here at the lower left, with the phosphorylation of ADP to form ATP. You're coupling electron transport with the phosphorylation of ADP to form ATP. Those two couplings, electron transport and the phosphorylation of ADP, together is called oxidative phosphorylation. And thereby you're making ATP. Now to finish off the straightforward biochemistry, what happens is once the ATP is formed in the matrix, it doesn't sit there. There is an antiporter system, and what the antiporter does, it transports ATP out and ADP comes in. ATP goes out, and ADP comes in. So it's an antiporter system. And then the very last of the straightforward biochemistry is what regulates oxidative phosphorylation? What regulates it? And the answer is it's ADP. That ADP is sometimes called respiratory control. When ADP levels are very high, oxidative phosphorylation goes faster and faster and faster. Okay, that controls the rate. All right, there it is. That is the straightforward biochemistry. Now, what do you need to know for in the integration with other disciplines? Well, as one example here of integration is physiology. Did you ever ask, why do you sweat when you exercise? You exercise, when you exercise, you sweat. So why is that? Okay, and the reason for that is, now just suppose this entire figure right here represents one muscle cell. And on your right side is where muscle contraction is taking place in the sarcoplasm. And there ATP is being consumed down to ADP as you exercise. And so now, as you start to exercise, because ATP is being consumed down to ADP, ADP levels start to rise in the sarcoplasm. And then ADP comes rushing back into the mitochondria to get reloaded with energy to form ATP. ATP comes rushing back out to do muscle contraction. ADP comes rushing back in just like a recyclable battery. ADP comes rushing back in to get reloaded with energy to form ATP. ATP comes rushing back out to do muscle contraction. So you can imagine, as you start to exercise, ADP comes in, ATP comes out. ADP comes in, ATP comes out. And remember, 50% of the energy is captured to make ATP, but the other 50% of the energy is given off as heat. And all that 50% of the energy given off as heat is why you sweat when you exercise. The massive quantities, all that perspiration, all that sweating that goes on is that 50% of the energy given off as heat. It's also the same principle, is that on a very cold day, for example, if you're in Chicago in January or February and you go out to the Miracle Mile and you expose yourself, and what are you going to do? When you expose yourself in a very cold day, you're going to shiver. You're going to go, man, this is cold. It's cold. I'm shivering here. What's all that shivering about? Because you're doing muscle contraction. And when you do muscle contraction, your 50% energy is given off as heat, and that heat is trying to maintain your body temperature at 98.6. That's why you sweat, and that's why you release it as energy, keeping the body temperature at 98.6. So that's biochemistry and physiology. But now here comes biochemistry and pharmacology. There are many drugs that affect oxidative phosphorylation, and the most important ones are listed here on the right. For example, barbiturates. High dose barbiturates can kill human beings. And also there is an insecticide called rotenone in toxicology. And rotenone, as well as barbiturates, what they do is, what you need to know is that they bind to complex 4. By binding to complex 4 does not work and will not transfer its electrons to coenzyme Q. And if it does not transfer to coenzyme Q, electron transport will not flow, and therefore no proton gradient, and therefore no ATP, and therefore you can die under that circumstance. Now you might say, well, what about the bypass reactions? Aren't they bypassing complex 1 to make some ATP? Yes, that's true. But that's not enough ATP. Remember, most of the electrons are in NADH, not FADH2. So yes, you can bypass, make a little bit of ATP, not enough. And therefore, that's how barbiturates kill, as well as rotenone. So if they ask you, you say they bind to complex 1, preventing electron transport to the coenzyme Q, therefore not enough ATP being made. Now, the one I want to spend some time on, because this is always on the boards and talking to medical students over the years, is cyanide. The boards hammer medical students regarding cyanide. Now, everybody knows cyanide kills. Now, how does cyanide kill? And what you need to know is that cyanide loves ferric iron, iron in the +3 state. Now, what happens is in complex 4, a little channel is open, and that channel enables cyanide to come into the channel. And when it comes into the channel in complex 4 only, it sees that ferric iron and binds to the ferric iron and never lets go. The ionic bond is so tight it never lets go. And so therefore, let me ask you this: what happens to electron transport when cyanide binds to ferric iron in complex 4? What happens to electron transport? It stops in its tracks. Why? Because electrons cannot go into that iron +3 to form iron +2 because it's bound to cyanide. So electron transport stops. Everything starts to back up. Electron flow stops. And if electron flow stops, no proton gradient, no ATP production, and you're dead. That's how cyanide kills. You must know that cyanide loves ferric iron in complex 4, and thereby stopping electron transport in its tracks. All right, and that's biochemistry with a killing mechanism of cyanide. Here comes the pharmacology. Now, in pharmacology, cyanide poisoning, there's a number of different drugs that can be used to treat cyanide poisoning. What's one of the major drugs used to treat cyanide poisoning? Let me see your answers here. What's one of the drugs used to treat cyanide poisoning? Let me see your answers here. Okay, now the thiosulfate, we'll talk about that in just a minute. But that's right. Angium, you said amyl nitrate. It's not nitrate, it's nitrite, like Daniel and Miranda and Lucas mentioned over here, Samantha now. It's nitrite, not nitrate. That's why I want you to use the public chat pod. Give answers, because if you give the wrong answer, you never forget it. It's not nitrate. It's nitrite is the antidote. And so now, why do you give nitrite? Why do you give nitrite? Let me see your answers here. It's amyl nitrite is what you usually use, amyl nitrite. Why? Because the pharmacology person told you to, or do you know why you give amyl nitrite? Not steals ferric iron, no. And the reason is because nitrite is an oxidizing agent. I know medical students hate that term, but nitrite is an oxidant, oxidizing agent. In other words, it takes electrons. And what's it take electrons from? It takes electrons from hemoglobin. So where iron is in the +2 state. And when you administer nitrite pharmacologically, nitrite is because it's an oxidizing agent, takes some electrons from the ferrous iron in hemoglobin, and therefore you're purposely making methemoglobin. Purposely making methemoglobin. Now, why are you doing that for? And the reason is cyanide doesn't care. Cyanide doesn't care where the ferric iron is located. Cyanide just wants to go to bed with ferric iron. It doesn't care. So by probability, because cyanide is in the bloodstream in cyanide poisoning, because methemoglobin is present in the bloodstream as well in the red blood cells, cyanide will see this ferric iron in methemoglobin first. It'll bind to it and sit there for 120 days, at which time you turn over the red blood cell and you pee out ferric cyanide into the urine. So you're simply tying up the cyanide in methemoglobin because it sees it first by probability. And you're trying to protect the ferric iron in complex 4, which is inside cells. So it's just simply the cyanide in the blood will see methemoglobin first before it comes into the cell and C is complex 4 ferric iron. That's why. That's why you give nitrite. Now, how much nitrite do you administer? I mean, that's what they do in pharmacology. That's why, I told you this earlier. That's what the pharmacologists, they tell you how much nitrite to administer, because after all, you don't want to give a whole kilo of nitrite. If you do, all of your hemoglobin would be methemoglobin, and you'd be dead for another reason. You couldn't carry oxygen anymore. So how much nitrite, how long does it last, blah, blah, blah. We have to give those pharmacology people something to do. And so otherwise they whine about everything. So the idea is but the biochemical basis of the pharmacology is right here. There it is. All right. Now, what's another antidote? Some of you said sodium thiosulfate, which is true. Now, why do you give sodium thiosulfate? Why do you give sodium thiosulfate? Because a pharmacology person told you to, or do you know why you give thiosulfate? Why do you give thiosulfate? To dispose of cyanide bound to methemoglobin? No. Good try. No. And the reason is, there we go, Purnot, that's it. And Andrea, that's right. And the reason is because chemically, thiosulfate converts cyanide to something called thiocyanate, which turns out to be less toxic than cyanide. It's still toxic, but less toxic than cyanide. And so, but the beauty of thiosulfate is it works very rapidly. Within seconds, the cyanide will convert to thiocyanate, which turns out to be less toxic. Now, in pharmacology, what's another antidote for cyanide poisoning? What's another antidote? You could use nitrite, you can use thiosulfate or some combination. That's right, Takir, that's right, Ricky, Daniel, and that's B12, and that is hydroxycobalamin. Now, why does B12 work? Because B12 has cobalt, and cobalt binds cyanide. Remember the bacteria flora, when they make B12 for me and you, they make it as cyanocobalamin. The cyano group is already there. It's showing you that cobalt binds cyanide. And then after you absorb the B12, you remove the cyanide enzymatically, but the amount of cyanide is trivial, not much. And so because the cobalt loves cyanide, if you administer orally hydroxycobalamin, it will bind the cyanide and prevent it from binding complex 4 ferric iron. There it is. There is the biochemistry behind the pharmacology, behind the pathology of cyanide poisoning. And Dr. Barone, next week, is going to be talking about the same thing. And when he starts talking about this in his beautiful lectures, you're going to say, oh, yeah, I know what he's talking about. Because we heard this already in biochemistry, and Dr. Barone will talk about it from a pathology point of view. The beauty, how this stuff works. All right, now that's cyanide. Now, carbon monoxide. Everybody knows carbon monoxide kills. Now, how does it kill biochemically? It's just the reverse of cyanide. Carbon monoxide loves ferrous iron, iron in the +2 state. Remember in complex 4, when an electron comes to the iron +3, the valency goes down to +2. And before it gives the electron to the oxygen, if carbon monoxide happens to be around, carbon monoxide will bind to that ferrous iron, preventing the electron from going on to the oxygen. Now, I know some of you are going to say, hey, carbon monoxide binds to ferrous iron in hemoglobin. That's true with very high affinity. That's true. But again, carbon monoxide doesn't care where the ferrous iron is located, either in hemoglobin or in complex 4. It's going to bind to it. In this case here, with complex 4, stops electron transport. You cannot make ATP, and that's part of the killing mechanism. All right, now a question for you. What is the antidote for carbon monoxide poisoning? The antidote in pharmacology or whatever. What's the antidote for carbon monoxide poisoning? Everybody knows this one? That's right. And that is hyperbaric oxygen, pure oxygen. And why you give pure oxygen? Because because carbon monoxide will bind to ferrous iron in hemoglobin, for example, and when pure oxygen is around, it displaces the carbon monoxide from the iron. So just simply reversible removal of the carbon monoxide from the iron. All right, high yield stuff. Now moving on, there is a toxic macrolide called oligomycin. And what oligomycin does and why it can kill me and you is, see this opening of the F0 channel? Just put a big X there. And by blocking the entry of the protons into this F0 channel, they cannot make ATP and you will die. And if they ask you how does oligomycin kill, you answer it prevents, put a big blockage between of the F0 channel, and the protons cannot come into the F0 and the F1 compartment, therefore no ATP. Now at the bottom of the figure are the notorious uncouplers. And one example is the one I want to talk about here. One example of an uncoupler is dinitrofenol, DNP, 2,4-dinitrofenol. And what DNP is, it's a weak base. And because it's a weak base, it picks up protons. So when it's administered, what happens is DNP will pick up the protons on the outside. And because DNP is permeable to the membrane, dinitrofenol will pick up the protons on the outside and goes right through the inner mitochondrial membrane and deposit those protons into the matrix, dissipating proton gradient. All right, now let me ask you this. Do electrons flow in the presence of dinitrofenol? Do electrons still flow? Yes or no? Are they still flowing? Is there any blockage of electron transport? And the answer is they still flow, yes. Electrons are still flowing onto the oxygen. Now because the protons, so as electrons are flowing, they're pumping the protons to the outside. But DNP picks them up and brings them right back into the matrix. So there's no proton gradient. Electrons are still flowing, but no proton gradient. And so as a result, you don't make enough ATP in the presence of an uncoupler. And that's what's going on. That's what the word means, uncoupler. You're uncoupling these two words. You're uncoupling oxidative, that's the electron transport. It's still going on. But you're uncoupling it from the phosphorylation of ADP to form ATP. You can't phosphorylate ADP if there's no proton gradient. So electron transport is still going on, but no phosphorylation of ADP. All right, now that's what uncouplers do. Like high-dose aspirin does that, as well as dinitrofenol. All right, now I got a little oral vignette. Get ready with your answer. Suppose they say to you that you are in the clinics, and in your clinics comes a teenager, and the teenager is very sick. And what happens is, what it turned out is that this teenager was on the local high school wrestling team. And you know, a lot of wrestlers in high school, they want to lose weight very quickly. So what the wrestler did was he went on the internet and purchased dinitrofenol. And the reason is because by taking dinitrofenol pills, that because you're uncoupling electron transport with the phosphorylation of ADP, because you're not making enough ATP, ADP levels in the cells, in the muscle cells, are going to be very high. ADP levels will be very high because you're not making enough ATP. And so because it's very high and because it controls the rate, then the electron transport will go faster and faster and faster, and you'll burn a lot of fat because you're doing a lot of oxidative metabolism. You'll burn a lot of fat and you will lose weight. So one more time. So the theory is, if a high school wrestler, because he wants to lose weight very rapidly, he takes, this wrestler went on the internet and purchased some dinitrofenol. with the idea being that if he takes DNP, you're not going to make enough ATP. ADP levels are very high, controlling the rate, making this go faster and faster and faster electron transport, and then more oxidative pathways are kicking in to make more, trying to make more ATP, and you will lose weight. Now suppose this wrestler, and why he became sick is, so he went on the internet and purchased dinitrophenol. It came the next day by FedEx. But instead of popping one pill, he popped a half a dozen pills. So therefore, they rush him to you as a doctor, and he's very, very sick, and he could die. Here comes the question from the boards: what could he die of? But why is it bad to take an uncoupler? Because you could die because of what? That's right. Because he can die of hyperthermia. Hyperthermia. That's right. Well done. And now, why hyperthermia here? And the reason is because, remember, when electrons are flowing to the oxygen, and then you're forming a proton gradient, and 50% of the energy is captured to make ATP, and 50% of the energy is given off as heat. But now, as the protons go back into the matrix, you're not capturing that 50% of the energy to make ATP. And what's happening here is 100% of the energy is given off as heat. And all that extra heat is causing massive hyperthermia. And the temperature can rise to 103, 104, and so on. And you could die by taking too much dinitrophenol. And if they ask you why, you say because of hyperthermia here. All right, there it is, how the biochemistry integrates with the pathology. Now another item here, and that is in immunology, when you talk about all those fevers that go on in the flu symptoms. When you have the flu, you have a fever, or in malaria, you can have a fever. And now, why is that? And because a lot of these cytokines that are being generated, what causes the fever in the flu or in malaria? What causes the fever are uncouplers in your cells. And so you're uncoupling electron transport, and then extra energy is given off as heat and causing the fever that goes on with the flu as well as in malaria. All right, one last item here, just to show you how important this is, because it integrates with pathology, immunology, pharmacology, and so on. But now, let me ask you this, in physiology and so on. Let me ask you this. In malaria, or anybody who has a massive fever, besides the elevated temperature, what is another characteristic of a high fever? What's another characteristic physiologically of a high fever besides the elevated temperature? Shivering, that's true. You shiver, that's true, because you're losing the energy and the heat. But what's another characteristic? Yes, shivering. What's another characteristic? Sweating, that's part of the shivering. Anjum, there we go. That's right. What's the other characteristic? Here it is. You ever know anybody who has malaria? They are hyperventilating. Why are they hyper? And the respiratory rate, as Anjum mentioned, is very high. Why are they hyperventilating? You're looking at the answer. You're looking at the answer. Why, when you have an elevated temperature, you hyperventilate? Because when electrons are flowing faster and faster and faster with uncouplers. So you're running electron transport very fast because ADP is elevated. Electron transport is going faster and faster and faster, and you got to ask, where do the electrons go? And you say they're going on to the oxygen. And so therefore you got to breathe in more oxygen, and therefore you hyperventilate. You're breathing in more and more oxygen to suck up those electrons in the electron transport chain. Amazing. But there it is, how the biochemistry explains the physiology, which explains the pathology, explains the immunology of a high fever. Why you hyperventilate, why you chill and sweat and so on, and major stuff. There it is. That's right. Never thought you thought biochemistry. Who cares? Who cares about biochemistry? You got to understand because it explains it all. All right. So we're done now with chapter 13. All right. Now this last chapter of carbohydrates is glycogen, major stuff. Get ready for a lot of path and endocrinology. Then after that, we're going to do gluconeogenesis. Be ready for a lot of stuff there. And pharmacology. And then after that, we're going to do the hexose monophosphate shunt. And wow, be ready for a lot of pathology in that pathway. So again, chapter 14 is very high yield stuff. Okay, now glycogen. Okay, now glycogen in this on the first figure here on glycogen. Now on page 200, everything you need to know about the straightforward glycogen synthesis and glycogen breakdown is right here on page 200. All the other pages, don't worry about glycogen synthesis and breakdown other than what's on page 200. As long as you understand this figure right here, you have everything, all the knowledge about the straightforward glycogen synthesis and glycogen breakdown on this single page. I'm boiling it down to what you need to know. And on this figure, major integration with endocrinology here. All right, now let's go through the straightforward biochemistry synthesis on glycogen synthesis on the left. Then after that, we'll do glycogen breakdown on the right. All right, so now the idea is, when do you make glycogen in glycogen synthesis? And you're all going to say, well, you make glycogen when you eat a high carbohydrate meal. So what happens is glucose then will enter your cells, and when glucose enters the cells, there is the enzyme called hexokinase in all cells, and in the liver there's the enzyme glucokinase, and what it does, it uses the terminal phosphate of ATP to stick that phosphate onto glucose, making glucose 6-phosphate as the product. And remember, the function of the phosphate on the glucose is to trap the glucose in the cell. Nothing new. We talked about that yesterday here. Now, in the liver and in the muscle cells, primarily. Now, in other cells, the glucose 6-phosphate can be burned in glycolysis or whatever. But in the liver and the muscle, to make glycogen, there is a low-yield enzyme. Don't worry about it, not even shown here, that converts glucose 6 to glucose 1-phosphate. No big deal. And then there's another enzyme, not even shown here, low yield, that's going to condense glucose 1-phosphate with UTP. And it activates the glucose to form UDP glucose as the product. Now remember, we talked about it yesterday in galactose metabolism. UDP glucose is a glucose donor. Anything in the body right now that has glucose in it, like glycogen coming up here, where did the glucose come from? Donation by UDP glucose, the high energy activated form of glucose. Now once UDP glucose is formed, we now see the major enzyme, the top enzyme of glycogen synthesis. You can't ask for a better name. Glycogen synthase synthesizes glycogen. And what does the enzyme do? It uses multiple UDP glucoses, and one at a time, it takes the UDP glucoses, and one at a time takes the glucose off and puts them onto glycogen, and glycogen will grow. Glucose alpha 1,4 glucose, alpha 1,4 glucose, alpha 1,4 glucose in linear arrangement. And then there's an accessory enzyme. You can't ask for a better name. And it's called branching enzyme. And what does the branching enzyme do? As the name implies, every so often it puts in a branch point of glucose in alpha 1,6 linkage. And these two enzymes are working together. The dominant enzyme is glycogen synthase. The accessory enzyme is branching enzyme. These two enzymes work together to make highly branched glycogen. And what you see here are 2 dimensions of glycogen, but in fact it's 3 dimension. Right in the middle is a small peptide called glycogenin that's low yield. But the idea is glycogen is highly branched, mostly alpha 1,4, but every so often alpha 1,6. Now in your book, as well as other books like First Aid, they talk about the mechanism of branching enzyme. Forget about it. They're not going to ask about the mechanism of branching. They're going to ask about the pathology of branching enzyme, which comes up on the next figure. So don't worry about the mechanism. Make life simple for yourself here. Now, why is this enzyme glycogen synthase highlighted in the red box? And the reason is the integration with endocrinology. Now, you notice here, you could predict. You don't have to memorize anything. If you were asked what hormones turn on glycogen synthase, and you would predict it's got to be insulin in the liver and the muscle turn on glycogen synthase, what do you think turns off glycogen synthase? Not even shown on this figure. What turns off, in case the boards ask you, which of the following hormones turns off glycogen synthase? What makes sense? That's right, you would all say glucagon, right? You would all say glucagon turns it off. Now, how does insulin turn on the enzyme? How does glucagon turn off the enzyme? You already know the mechanisms. That was discussed last few days, how glucagon and insulin work. No need to go and review that again. That's already been reviewed. All right, now once glycogen is made, that's glycogen synthesis and its regulation at the hormone level. There it is, how simple it is. Not hard, nothing to panic about. Just master what we just covered. Now, once glycogen is made, it doesn't sit there. When you need that glycogen, when you have to break down the glycogen, here comes the top enzyme of glycogen breakdown. And that enzyme, major enzyme in pathology coming up after the break. But this enzyme is called glycogen phosphorylase. Now, what does a phosphorylase do? By definition, the surname phosphorylase. Notice it's not a phosphatase. It's a phosphorylase. And what the enzyme does, by definition, it uses inorganic phosphate to cleave sugar bonds. So the product is glucose phosphate, not glucose. It's not a hydrolase. Hydrolase uses water to cleave sugar bonds. It's a phosphorylase that uses inorganic phosphate to cleave the glucose off as glucose 1-phosphate as the product. So this enzyme is cleaving off the alpha 1,4 bonds, and then there's an accessory enzyme. You can't ask for a better name. It's called debranching enzyme, and every so often it cleaves off the alpha 1,6 linkages. So these two enzymes, again, work in harmony. They work together. The dominant enzyme is glycogen phosphorylase, which uses inorganic phosphate to cleave the sugars off. And then the accessory enzyme is the debranching enzyme. Now, again, in your book, as well as in First Aid, they talk about the mechanism of debranching enzyme. Forget about it. I ain't going to worry about the mechanism. What you need to worry about is the integration with endocrinology. Now, here it is. You see the list here, but there's no reason to be here because you would predict if the board asks you, if they ask you which of the following hormones turn on glycogen phosphorylase, you would predict, and rightfully so, glucagon in the liver turns on the enzyme, epinephrine in the liver and the muscle also turn on the enzyme. Now, I'm not going to tell you how glucagon and epinephrine turns on the enzyme because I don't want to bore you to death because we talked about it several times over the last several days. And if I tell you how these two hormones work, you're all going to be bored. You're going to be, and Natalie's going to say, man, I'm bored with all this stuff and I'm a very sensitive guy. And so I'm not going to tell you how these hormones work. But if I was going to tell you, what I would say is how epinephrine and glucagon turn on glycogen phosphorylase. What they do is they bind to the beta receptor on the respective tissues. By binding to the beta receptor, the receptor undergoes a conformational change, which then in turn turns on GS, stimulatory G protein. And then alpha S of GS binds GTP. Alpha S in the presence of GTP in turn turns on adenosylase to make cyclic AMP. The cyclic AMP then turns on protein kinase A. Protein kinase A then phosphorylates the phosphorylase, turning the enzyme on, and you break down glycogen. But I'm not going to tell you that. I'm not going to tell you that because I don't want to bore you to death, so I'm not going to tell you. But if I was going to tell you, that's exactly the verbiage that would come out of my mouth. Exactly that verbiage would come out of my mouth. But I'm not going to tell you because I don't want you yawning at me, and I'm a sensitive guy. All right, now moving on. Now it says over here that AMP allosterically, not hormonally, allosterically turns on, in the muscle only, turns on glycogen phosphorylase. Now let me ask you this. Does that make sense to you? Do you think you have to memorize that? On board exams, if they say to you, which of the following molecules allosterically turns on phosphorylase in hard working muscle? If one of the options is AMP, does that make sense to you? It should. Because when you exercise, ADP is being consumed down to ADP, which consumes down to AMP. And AMP levels in the hard-working muscle rise. And then AMP in hard-working muscle turn on glycogen phosphorylase. You break down glycogen to glucose. And remember, AMP turns on PFK1 in glycolysis. Remember all that? So AMP is generating the fuel to burn and turn on the pathway of glycolysis. It's beautiful how this works. You can memorize all you want, but unless you understand why, oh yeah, this is beautiful, how AMP turns on glycogen breakdown as well as glycolysis to generate ATP when you exercise. So there's no need to memorize that AMP turns on the enzyme. Because on board exams, if they ask you which of the following molecules turns on the phosphorylase, if option A is let's say plutonium-238, don't put that as a correct answer because it doesn't make sense. What makes sense is AMP. All right, there it is. All right, now what do you think turns off in endocrinology? What hormone makes sense to you turns off glycogen phosphorylase? What makes sense to you? What turns it off? That's right. It's got to be insulin. Insulin turns off glycogen phosphorylase. Now you know the mechanism, how insulin works. So this, it says insulin turns off the phosphorylase. I'm not going to tell you how insulin turns off glycogen phosphorylase because I don't want to bore you and you're all yawning at me. You think, he's boring me with this stuff. So I'm not going to tell you how insulin turns off the enzyme. But if I was going to tell you, what I would say is, what insulin does is it binds to its receptor, which then the receptor, it's a tyrosine kinase class receptor. And in the presence of insulin, the receptor dimerizes. And once it dimerizes, because it has tyrosine kinase activity, it autophosphorylates itself on the cytoplasmic side. That in turn attracts IRS1, which in turn attracts SH2 proteins, which in turn in the cytoplasm attract other proteins, and downstream activates a phosphatase. The phosphatase then dephosphorylates the phosphorylase, therefore turning the enzyme off in the presence of insulin in the bloodstream. But I'm not going to tell you that because I don't want to bore you to death. But if I was going to tell you how does insulin work, turn off the enzyme, that's the exact wording, that's the exact verbiage I would use. But I'm not going to tell you that because I don't want you yawning at me. But that's exactly what I would say. But I'm not going to tell you. So don't go asking me. Don't go using the Q&A pod and say, Dr. Erwin, Dr. Zeta, hey, please help me understand how does insulin turn off the phosphorylase. You guys don't worry because it's already been reviewed. So no need. You understand. You've mastered how insulin and glucagon epinephrine work. All right, now what we're going to do is we're going to come back to this page after the break. So let's take a 10-minute break, and then I will return. It's a minute to the hour, so let's take a 10-minute break, and I will be back. Welcome back, everybody. I was reading there. I see that Maritza's daughter got into the National Junior Honor Society. Well done. Congratulations. Very proud to see your daughter get in there like that. That's a very huge accomplishment. But I bet your daughter thinks and not memorizes, because if you think, you're going to do well. Anybody can do well. And I'm glad she's a thinker. All right, now what's coming up here is that's right, think. Just all you got to do. And Renaud, you say you're getting nervous for the exam. Nah, don't. Have faith in your ability. You could do it. Just all the information. We're giving you the information. We're giving you what you need to know. There's no reason to go reading other books and reading this and that. Just focus in on these figures in front of you right now. For example, understand this figure, be able to use the information, apply the information for vignettes and questions. We'll be practicing that at the end of this chapter as well, some vignettes. That's all you need to do. Don't go reading all kind of other garbage out there. There's a lot of garbage you don't have to know. Focus your attention on the high yield material. All right, now coming back to this figure here is that when you start breaking down glycogen when you start breaking down glycogen by activating the glycogen phosphorylase, and conjunction with debranching enzyme, we generate glucose 1-phosphate. And then the glucose 1-phosphate converts to glucose 6-phosphate. And then now here is a major item of location. What happens there is in the liver, there is a major enzyme. Get ready with that highlighter, called glucose 6-phosphatase. Look at the name of the enzyme, tells you what it does. Glucose 6-phosphatase. This is the enzyme that removes that phosphate group from glucose 6-phosphate, because it's named after that, and then thereby the glucose can now exit the liver and they go out there into the bloodstream. Now remember earlier I mentioned to you that both the liver and the muscle have about a half a pound of glycogen. But the liver is solely responsible for maintaining blood glucose, not the muscle. Even though the muscle has glycogen, the muscle is a very selfish and greedy organ. Whereas the liver is a very kind and wonderful organ, because the liver says, I am the united way of organs. And so when the community of organs need glucose, the liver shares, whereas the muscle does not share. And the reason is because the muscle lacks the glucose 6-phosphatase enzyme. It has the gene for it, but it puts the gene into heterochromatin, never to turn it on into RNA, into enzyme. So even though it has the gene, the muscle willingly turns it off because it's a selfish organ. It's all about me, the muscle says. Whereas the liver says, I got the gene too, but I'm going to put it into euchromatin in order to make RNA to make the enzyme so we can get the glucose out of the cell to try to maintain blood glucose values here. So again, the idea is so both of these organs have glycogen, but only the liver shares the glycogen, not the muscle here. All right, now if you understand this all right, now don't turn the page. Eyes up, don't go turning the page. All right, Daniel, you turn that page, don't go turning that page. All right, okay, don't turn the page. Okay, now, Maritza, you should be able to get this because your daughter can think and you can think too. Okay. All right. Now, when we turn the page, what we're going to see are the table of the glycogen storage diseases in pathology, and there are 6 of them. And don't go turn. I thought I heard somebody turning the page. All right. Good. Nobody turning the page. Now, when we turn the page, we're going to see the glycogen storage diseases. There are going to be 6 of those listed. And now at the top, type 1. Now in type 1 glycogen storage disease, first of all, what is the name of the disease? Don't go turning the page. Don't look at the answer. What's the name of the disease in which glucose 6-phosphatase enzyme in the liver is genetically defective? What's the name of the disease? That's right. It's called von Gurke's disease. Von Gerke's disease. All right, now in von Gerke's disease. So we're going to see this table. We're going to see type 1. We're going to see von Gerke's disease, the name. We're going to see the enzyme glucose 6-phosphatase enzyme is defective, and then we're going to see a column of the pathologies, the clinical manifestations. Now here it is. What do you expect? Because you're a thinking person. What do you expect? I almost gave it away. What do you expect must be the #1 hallmark of von Gerke's disease? If what I told you is correct, and it is, what do you expect to see as the #1 hallmark of von Gerke's disease? In fact, not expect to see it. You demand to see this #1 hallmark, or you want your money back, right? So what do you expect to see is the #1 hallmark of von Gerke's disease? Hepatomegaly is true, but what's the other hallmark? And that is severe hypoglycemia. Why? Because if this enzyme is defective, you cannot remove the phosphate group. The glucose 6-phosphate's trapped in the liver and never exits the liver. The blood glucose drops. Severe hypoglycemia. And there it is. Type 1. Von Gerke's disease, the enzyme glucose 6-phosphatase, and here it is, severe hypoglycemia. Look how you figured that out all by yourself. You figured that out. I didn't tell you. You figured it out. Why? You use your mind. That's how you knew the answer, because you thought of it. Because you didn't memorize that. It just makes sense. Is why you got the answer. Now, in addition, they have lactic acidosis. We'll talk about that later if we have time. And they have hepatomegaly. Now, a lot of you said hepatomegaly. That's the other characteristic, other hallmark. And the reason for that is because if glucose 6-phosphatase is defective, then the phosphate cannot be removed. Glucose 6-phosphate stays in the liver, and it's osmotic, drawing water into the liver. It backs up to the glycogen, which is osmotic, drawing water into the liver. There it is, hepatomegaly here. All right, now in addition, they have hyperlipidemia. Now this one's harder to figure out because we haven't talked about lipids yet. But the idea is, if you think about it, oh yeah, this makes sense too. How do you explain the hyperlipidemia? Again, it's a little bit harder to figure out, but you have to think. Why do you think they have hyperlipidemia? Why do they have hyperlipidemia? Now think about it. It takes a little bit of thought. Why do they have hyperlipidemia? To get a ride of glucose, they make fatty acids. No, good try. You would think, oh, it's because of the glucose making the fatty acids and triglycerides. No, good try. Fats metabolized as low glucose. No. Now here comes the answer. Remember, I told you this the other day. Is the way to understand metabolism, as an example right now, to understand metabolism and pathology, you always say is, what condition is the body in? And once you know what condition the body in, you ask what hormone or hormones are being secreted in response to that condition. And then what's the target organ of those hormones? And in that target organ, what is the key pathway you want to turn on or off? And what enzyme you want to turn on or off of that particular pathway. So here it is. Here is the perfect example of this, what I just mentioned. What condition is the body in? And you say, well, the condition in von Gerke's, the body is in hypoglycemia. How does the body respond to the hypoglycemia? Well, it sends out hormones. Like what? Like cortisol, like epinephrine, like ACTH, like norepinephrine, and so on, cortisol and so on. And what are those hormones going to do? A lot of these hormones will go to the adipocyte. And when they go to the adipocyte as the target organ, what pathway is going to be turned on? Triglyceride mobilization. And what's the top enzyme that breaks down triglycerides in adipocyte that's sensitive to all those hormones? The name of the enzyme that's sensitive to the hormones again. What's the name of the enzyme? Oh yeah, hormone sensitive lipase. HSL is turned on, triglycerides are mobilized, fatty acids pour into the blood. Hyperlipidemia. It makes sense. No mystery here. No mystery. There it is. There it is. It makes sense. Because you always say, what condition is a body in? And how does the body respond to that condition? And then what's the target organ? And then what's the target pathway in that organ? What's the key enzyme of that pathway of that organ? It just makes sense. Think of it that way, things start to fall into place. Trust me, that's the best way to understand this. All right, now they have other characteristics as well, but the main one is severe hypoglycemia accompanied by the hepatomegaly. All right, now let's continue. Now, in my opinion, you got to know all 6 of these. You should know the name, the type, as well as the name of the disease. Now, the next one I'm talking about is Pompe's disease. Now, in Pompe's disease, it's where a lysosomal alpha-1,4 glucosidase is genetically defective. Now, let me say something about this enzyme. No one knows truly what the enzyme really does. What's the function of the enzyme? Notice its location. It's located in the lysosomes. Remember, glycogen is made and broken down in the cytoplasm. So what's this enzyme doing in the lysosomes? No one knows. It's believed that when glycogen gets broken down to a little stub, the glycogen is finished off in the lysosomes. But that's just a guess. No one knows the true function of this lysosomal enzyme. But notice, here's an enzyme that no one really knows what it does. And if you don't have it, look at this. Death by 2 years of age. Incredible. Here's an enzyme no one knows. If you don't have it, death by 2 years of age. Now the idea here is the main characteristic, the main hallmark of the disease, cardiomegaly. If you see cardiomegaly on national boards, you automatically think they might be asking me about Pompe's disease. You want to see cardiomegaly in Pompe's? There it is right here. Look how massive the heart is in Pompe's disease. That's why they don't live very long. The heart is so massive that the body cannot accommodate that massive organ called the cardiomegaly of the heart here. It just takes up too much room. That's why they don't live very long. All right, now continuing on, the next 2 to talk about are Corey's and Anderson's. Now, Corey's is where the debranching enzyme is defective. Anderson is where the branching enzyme is defective. There's an easy way to remember what enzyme goes with what name. What enzyme goes with what name? All you need to know are the 4 letters of the alphabet. A, B, C, D. Here it comes. A, Anderson. B, branching. C, Corey. D, debranching. A, B, C, D. Helps you understand what enzyme goes with what name. Now, the important feature here is that if there's something wrong with branching or debranching enzyme, then you look to see, is there something wrong with the glycogen structure? In other words, a biopsy was taken of the muscle or the liver or whatever, and they looked at the glycogen, and they found out it was an abnormal glycogen structure. Notice with the other ones, they have normal glycogen structure, but with Corey's or Anderson's, they have an abnormal glycogen structure. Now, don't look in the purple box. Don't look there. Okay? Just give yourself a chance to get to the right answer. If it's Corey's, if it's Anderson's disease, where the branching enzyme is defective, what do you expect the glycogen to look like? It's going to be abnormal in structure, but what do you expect the glycogen to look like in Anderson's disease, where the branching enzyme is defective? That's right. You expect it to look linear. That's right. It looks like starch. It looks like starch because it's linear glycogen. Okay, now if it's D-branching enzyme is defective, what do you expect the glycogen to look like in case they tell you on the boards? They say a biopsy was done, they looked at the glycogen, and it was abnormal, and it had this feature. What do you expect if D-branching enzyme is defective, the glycogen to look like? And you would say, well, glycogen would have all these little stubs, small stubs throughout the glycogen particle, and these small stubs are single glucose residues at the alpha-1,6 branch points, because the alpha-1,6 glucoses cannot be removed at the branch point because D-branching enzyme is defective, and that's CORS. Again, it just makes sense. There's not hard here. Don't go memorizing. It's logical. It just makes sense. Now the bottom two are McArdle's and Her's disease. Now notice here in both cases it's the same kind of enzyme, the phosphorylase. But notice location, location, location. McArdle's disease is where the muscle phosphorylase is defective, sometimes called the myophosphorylase. Same thing, the muscle phosphorylase is defective, whereas in Her's disease it's the liver phosphorylase is defective. And again, easy way to remember what enzyme goes with what name. M. McArdle, M. muscle. H. Her's, H. hepatic phosphorylase. So again, it's the same kind of enzyme, but the locations are much different. All right, now don't look at the clinical manifestations here. Now, if it was McArdle's disease, if you get a vignette on board exams and it's McArdle's disease, what do you expect to see on the national board in terms of a vignette? And you would answer by saying, well, if it's McArdle's disease, you expect to see something like this in a vignette. You would say a child is going to the playground to run around with the playmates. As a child runs around, the child starts to complain of muscle pain, muscle cramping, muscle weakness. So how would you explain that if it's McArdle's disease? You explain that by saying, well, as a child runs around, the child's exercising, therefore the child is secreting epinephrine. Among several tissues epinephrine will go to, one of them is the muscle cell. And so it binds to the beta-adrenergic receptor in the muscle cell, and then it stimulates protein kinase A downstream to phosphorylate the phosphorylase in the muscle. But if the phosphorylase is defective, they can't break down glycogen to get the glucose to make ATP to do muscle contraction. Therefore, muscle cramps and weakness upon exercise. It makes sense. Now, they also have myoglobinuria because some of the muscle cells will die under those circumstances, but there it is. Not hard at all. All right, now let me ask you this. What other disease and pathology sounds very similar to the vignette I just mentioned? Because on board exams, they love it when two diseases or more sound very similar. They're going to want to know, how do you know? What is a differential diagnosis? What other disease sounds very similar to what I just mentioned here? Now, if you're thinking, and you said there, Duchenne's muscular dystrophy or M. gravis, something like that. No. Because those patients with muscular dystrophy, for example, they would have muscle problems just by walking. The key here is exercise. And exercise means an epinephrine activated pathway is not working like it should. And what other disease sounds very similar? And the answer is myopathic carnitine acyltransferase deficiency. It's a fatty acid oxidation defect. Now, don't worry about it tonight. Tomorrow, we're going to get into the fatty acid oxidation pathways and the pathologies, and there you will see myopathic CAT deficiency is a fatty acid oxidation defect. They can't burn fatty acids properly. Not enough ATP, therefore muscle weakness upon exercise. So again, they're going to love this because they're going to want to know the differential diagnosis. Myopathic CAT deficiency. That's like VO says there. There it is. That's right. Also could be MCAT deficiency. Very similar. But those are fatty acid oxidation defects. Now the last disease here is Her's disease. where the hepatic phosphorylase is defective. Now, so again, in McArdle's disease, they have an increase in glycogen in the muscle only. So in McArdle's disease, now let me ask you this. In McArdle's disease, do you expect to see, without looking here, do you expect to see hepatomegaly in McArdle's disease? Yes or no? Do you expect to see hepatomegaly in McArdle's disease? No. Do you expect to see hypoglycemia? Do you expect to see hypoglycemia in McArdle's disease? Yes or no? And the answer is no. Why? There's nothing wrong with the liver. The liver is fine. The liver is processing its glycogen normally. This is a muscle problem. Now, Her's disease is a liver problem. So we have increased glycogen in the liver only. So as a result, they end up with hepatomegaly because the glycogen can't be broken down properly. Now, don't look at the other clinical manifestations. Now, yes or no. I'm looking for a yes or no answer. Without looking, now if you look, don't answer. If I told you in Her's disease, they have, in which the liver phosphorylase is defective, if I told you they have hepatomegaly, and you would say, yes, they have hepatomegaly. If do they have hypoglycemia? Do they have severe hypoglycemia? If they can't break down glycogen in liver, do they have severe hypoglycemia? And without looking here. And the answer is no. They have no hypoglycemia, mild at worst. They have hepatomegaly, but no hypoglycemia. How do you explain that? How would you explain that? They can't break down the glycogen, yet they're maintaining blood glucose. What does that tell you before I tell you? What is that telling you, because you're a thinker? Well, if the blood glucose is normal, yet they can't break down glycogen, what's that telling you? That should be telling you there's got to be another pathway kicking in to maintain blood glucose. And sure enough, there is. Remember, there are two pathways to maintain blood glucose. One is glycogen, and the other is gluconeogenesis. Both of them generate glucose 6-phosphate. Remove the phosphate to generate the glucose to get out of the liver, to get into the bloodstream. Now, the idea is, in von Gurke's disease type 1 and in Her's disease type 6, notice both of them have hepatomegaly, but in one case, severe hypoglycemia. In the other case, no hypoglycemia. How do you explain that? And the answer is simple. At the bottom of the figure here, which arrow is defective? Here it is. In Her's disease, which of these three arrows here is defective in Her's disease? And the answer is, it's got to be this arrow there. Notice how gluconeogenesis is maintaining blood glucose. No problem. But in von Gurke's disease, which arrow is defective? And clearly, it's that arrow right there. And now notice how in von Gurke's disease, why is it severe hypoglycemia? Because neither glycogen nor gluconeogenesis can maintain blood glucose. There it is. It makes sense. There it is. Not hard. If you're a thinker, not hard. Now, here it is. Suppose they say to you, and get ready with your answers. Suppose they say to you that the patient has hepatomegaly, but the blood glucose, is 3.5 millimolar blood glucose. Which disease do they have? Is that von Gurke's or is that Her's? They have hepatomegaly and the blood glucose is 3.5 millimolar. And that's von Gurke's. Remember, 3.5 millimolar. That is severe hypoglycemia. Remember we talked about that with the transporters. That 3.5 is physiologically low as you will get, maybe 3.4, 3.3, but 3.5 millimolar is severe hypoglycemia. Now, if I said, remember the lower end of normal is 4 millimolar, and so this is 3.5, that's very severe hypoglycemia. Now, using the other units, milligrams per deciliter. Suppose they say to you the patient has hepatomegaly and the blood glucose is 72 milligrams per deciliter. What disease do they have? 72 milligrams per deciliter. That's right. That's the HERS, because the 72 millimolar is on the low end of normal, and so still low, but they are the low end of normal. That would be a mild hypoglycemia, and that would be HERS disease. So that blood glucose is so critical to understand. You need to know the blood glucose in millimolar units and milligrams per deciliter units. There it is. You save valuable time. So again, blood glucose is 90 milligrams per deciliter in terms of absolute value, but in range, according to the boards in their table, it's 70 to 110. It's considered normal. So 72 milligrams per deciliter is a low end of normal. Okay, now what's coming up next is the pathway of gluconeogenesis. Here, now this figure here on page 206, now it looks complicated, but it's really not that bad. And the reason why, because there's two pathways here. On the right side is glycolysis, on the left side is gluconeogenesis. So even though with all these arrows it looks formidable, it's really not that bad. Okay, now the idea is that in gluconeogenesis and glycolysis, now it turns out that glycolysis has 10 steps, 3 of which are irreversible. And so the pathway of gluconeogenesis, they use the same 7 reversible enzymes, but the 3 enzymes that are irreversible, gluconeogenesis has a different set of enzymes to reverse those irreversible steps. Now, the important feature here is that if you were asked what tissues in the body make glucose, you say the liver. But according to a national board exam, according to a student, I should say, they told me on board exams, they asked, what's the other organ in the body besides the liver? The liver is the dominant organ that makes glucose. But what other tissue or organ can make glucose? And what is it? That's right. And you've got to know it because it's in their database of questions. That's right. It is the kidney. The kidney is a minor player for making glucose, but it still makes glucose. And they ask that on the national board. Be prepared here. Okay, now what I want to do here is on the right side is go through glycolysis in a nutshell once again. And now how does that compare with gluconeogenesis? On the right side, so when glucose comes into the liver in glycolysis, it sees the enzyme glucokinase. Because the surname is kinase, that means it needs ATP to work. Because ATP is being consumed to phosphorylate glucose, then it's an irreversible step, meaning gluconeogenesis has to have a different enzyme to reverse this. Now, once glucose 6 is formed, generates fructose 6, no big deal. And then it sees the top gun of glycolysis, and that is PFK1, phosphofructokinase 1. Because it's a kinase, that means ATP is being consumed. Because ATP is being consumed, that's another irreversible step, meaning gluconeogenesis has to have another step, another enzyme to go in the reverse direction. Now, before we proceed, just to review this one more time. When you eat a high carbohydrate meal, what's telling PFK1 to work again? Again, just without looking in your book, because we talked about this late yesterday. What's telling PFK1 to work when you eat a high carbohydrate meal? That's right, it was insulin. Because what happens is insulin turns on PFK2, which generates that product, fructose 2,6-bisphosphate. Remember all that? Which overrides any inhibition of PFK1. And so what turns it on? It is insulin here. All right, and now remember, now if I were to ask you, what other enzyme in glycolysis is turned on by insulin again? What other enzyme in glycolysis, without looking in the book, is turned on by insulin? What other enzyme? That's right, it's showing right here. It's glucagonase. So notice here, both enzymes that are turned on by insulin succinctly are shown right here again to help you remember. And if you're asked what hormone turns this off, you would say it's glucagon turns off the enzyme, and therefore you decrease fructose 6-bisphosphate, and you don't turn on the enzyme. And if I were to ask you, nothing new, if I were to ask you allosterically what turns off PFK1, you would say it's got to be ATP and citrate. Nothing new, nothing new. Now let's proceed. Now once fructose 1,6-bisphosphate is formed, after several reversible steps, low yield, we generate the highest energy compound in the cell, PEP, phosphoenolpyruvate. And then there's that last enzyme of glycolysis, pyruvate kinase, and what does it do? It takes the high energy phosphate from PEP and uses it to make ATP. Because the highest energy compound is being consumed, that's the third and last irreversible step. That gluconeogenesis has to have a different set of enzymes to go in the reverse reaction. So then we generate pyruvate. So there it is again, glycolysis in a nutshell. Wasn't hard, nothing new. But notice that 3 steps that are irreversible in glycolysis, these were the 3 steps that we in Kaplan Medical recommend you knowing for the national board. Okay, so now we're ready to do gluconeogenesis. Now to make glucose, the genesis of glucose in the liver, as well as the kidney to a minor player, you got to ask, where is the carbon coming from that the liver uses to make glucose? As we talked about yesterday, way back on page 163. One of the sources of carbon is pyruvate and lactate, same thing, just a hydroxyl group difference. A second source are the glucogenic amino acids that will funnel their way down to the alanine, which gets dumped into the blood. Alanine levels rise, which goes to the liver, and it converts it into pyruvate. Let me ask you this. Get ready with your fingers here to type. How many amino acids in the human body are glucogenic? I know some amino acids are both glucogenic and ketogenic, but how many amino acids out of 20 can you use to make glucose? You have a number from 0 to 20 to choose from. How many amino acids are glucogenic? Not 4, not 2, not 7, not 6, not 10, not 5. Yeah, there it is. Not 11, not 15. All over the place. But Gia did get it right, and Jung, there it is. And the answer is 18. 18 amino acids are glucogenic. And by default, the other 2 are solely ketogenic. And the easiest way to remember this is just remember the 2 ketogenic amino acids. And they are the 2 L's. Lysine and leucine are solely ketogenic. The 2 L's, lysine and leucine. And by default, all 18 other amino acids can be used to make glucose. They're a very easy way to remember that. Okay, now the third and last source of carbon is glycerol. Okay, now we're ready. We've accounted for where all the carbon is coming from to use to make glucose. Now, the hardest part of gluconeogenesis is reversing this seemingly irreversible step. How do we go from pyruvate to PEP? And it turns out we don't need 1. We need 2 major enzymes to reverse this seemingly irreversible step. And so what happens is the pyruvate goes into the mitochondria. And there, the first of the 2 critical enzymes is called pyruvate carboxylase. Now, what does the enzyme do? It takes the 3-carbon pyruvate and puts CO2 onto it using ATP to make the 4-carbon product called oxaloacetate. And notice what the enzyme needs to work. It needs ATP and CO2, and to do that, it needs biotin as the vitamin to make the coenzyme here. So it needs ATP, biotin, and CO2. Now you can remember these 3 by knowing that pyruvate carboxylase is viewed as an ABC carboxylase. The 3 letters of ABC help you remember what the enzyme needs to work. A for ATP, B for biotin, and C for CO2. The first of 3 First of three critical ABC carboxylases in the human body. And this one here is in gluconeogenesis. All right, now another reason why you need to know this enzyme. There's no pathology or pharm. Why you need to know this enzyme, actually there will be some pharmacology, but why you need to know this enzyme is because it's highly regulated, and it's regulated by acetyl-CoA. Now notice here how acetyl-CoA inhibits PDH, we talked about that yesterday, and how acetyl-CoA turns on pyruvate carboxylase. So now let me say something about acetyl-CoA turning on pyruvate carboxylase. Acetyl-CoA is the first of two examples in the human body of what's known as an obligate activator of an enzyme. Obligate activator of an enzyme. Now, what does that mean? Obligate versus allosteric activator. Now, most activators are allosteric. Now, what does that mean? Now, got to pay attention here. Now, what an allosteric activator of an enzyme is, it means the enzyme works all by itself, but it works better when the allosteric activator is present. An obligate activator means the enzyme doesn't work at all unless the obligate activator is present. In other words, pyruvate carboxylase is like the engine, and acetyl-CoA is the key to turn on the engine. So without acetyl-CoA, the enzyme never works, the obligate activator of the enzyme. It's only two examples in the human body. This is the first pathway that requires an obligate activator. Does anybody know? If you forgot, don't worry, we'll get to it later. Does anybody know what other pathway in the human body requires an obligate activator to turn on that pathway? Does anybody know what other pathway requires an obligate activator? Good try, not fatty acid oxidation. Good try, Muscan. Good try. No. Not the hectose monophosphate shunt. No. Rouge. No. Cholesterol. No. Not. There we go, Samanthony. And the answer is it's the urea cycle. We'll talk about on Friday. The urea cycle is the other pathway that requires an obligate activator. Obligate means mandatory, means required to be there to turn on the enzyme, therefore the pathway. Gluconeogenesis is one, urea cycle is the other pathway. All right, let's continue. Now this 4-carbon oxaloacetate, again, what are we trying to do? We're trying to reverse pyruvate going back to PEP. Now what happens is the OAA product in the mitochondria gets shuttled out of the mitochondria, low yield. And goes out into the cytoplasm. And there it sees the second major enzyme called PEP carboxykinase. And what does the enzyme do? It takes a 4-carbon OAA, splits off one of the carbons as CO2, using GTP to do that, and makes PEP as the product, the 3-carbon PEP as the product here. All right, now notice over here. Notice how this first enzyme uses energy, ATP, to put CO2 on. The next enzyme uses GTP to take it right back off. It's like your body can't figure out what to do with that CO2. But why you are doing this is driving the formation of the product here. Is by removing the CO2 gas, you're driving the formation of the highest energy compound in the cell. That's PEP. All right, now this enzyme, PEP carboxykinase, what hormones, you should already know this, what hormones turn on PEP carboxykinase? What hormones? Andrea, that's right, and Daniel, glucagon and cortisol. How would you know that? And you're all going to tell me page 78. Page 78. How many times have we talked about that in this class? Page 78, how glucagon and cortisol turn on PEP carboxykinase at the gene level. Remember the zinc finger receptor for cortisol. Remember the Krebs protein for glucagon. High yield stuff. All right, let's finish off this figure. All right, so now we have reversed using two major enzymes to make PEP. After several reversible steps, we generate fructose 1,6-bisphosphate, and then there is the major enzyme in gluconeogenesis called, oh, by the way, notice here how gluconeogenesis is energy requiring. To make that glucose, you need ATP and GTP to power gluconeogenesis. That's going to be so critical soon. Please remember gluconeogenesis costs energy. All right, let's continue. Now, once it backs up to fructose 1,6-bisphosphate, there is another major enzyme called fructose 1,6-bisphosphatase, and what it does, it pops off the phosphate group, generating fructose 6-phosphate as the product. Now, notice here in this figure, in the cytoplasm of the liver, there are two major enzymes of their respective pathways. PFK1, the major pathway of glycolysis on the right side. On the left side is fructose 1,6-bisphosphatase, a major enzyme of gluconeogenesis. It would make no sense that these two enzymes work at the same time. It would be futile. So you would imagine the enzyme on the left got to be highly regulated, because you don't want to turn on both enzymes at the same time. So if you were to choose a metabolite that turns off fructose 1,6-bisphosphatase at the same time turn on PFK1, well, you would say, well, why not choose fructose 2,6-bisphosphate under the command of insulin? And sure enough, it does. So insulin turns off gluconeogenesis by indirectly making this substance here. There it is. It's beautiful how this works. And now if you would ask what turns on this enzyme, why not choose ATP? ATP turns on the enzyme. Allosterically, ATP turns off glycolysis. Beautiful. This is how, notice here, how these two pathways never work at the same time, because they are reciprocally regulated. They never work at the same time, because both enzymes cannot work at the same time, because they have reciprocal regulation. All right, now to finish off this figure. Now the question becomes, now think big picture. In starvation conditions, when the liver, and when you haven't eaten anything for weeks at a time, you haven't eaten anything, no carbon coming into the mouth, yet the liver's making glucose. And we have now accounted for where the carbon is coming from to make glucose. But now, where's the energy coming from? Now think about this. You're starving, yet the liver needs energy to power gluconeogenesis. Where is the energy coming from when you're starving? Let me see your answers here. That's right. Where it's coming from, because you're saying, because these hormones are going to the adipocyte and turning on HSL. Triglycerides are being mobilized, and then the fatty acids pour into the liver. They go through beta oxidation to turn on gluconeogenesis and to power gluconeogenesis there. There it is. It makes sense. Now, let me show you here how, if you know a little bit about chemistry, it goes a long way in pathology. Now, let me ask you this. We'll talk about it tomorrow. Don't worry about it today. But tomorrow, we're going to be talking about beta oxidation. And then we'll get into the pathologies of beta oxidation. If what I'm telling you right here is correct, and it is, if somebody has a defect in pathology of beta oxidation, they can't burn fatty acids properly, what do you expect to see as the #1 hallmark of the disease in beta oxidation? If you have a beta oxidation defect of fatty acids, what do you expect got to be the #1 hallmark? That's right, severe hypo, not hyper, BO, it's hypoglycemia. Severe hypoglycemia. They can't make enough glucose. You got to know that, because that's what a lot of sudden infant death syndrome is about. An infant sleeping in bed in the crib, and they don't wake up. And if they have and it could be due to a beta oxidation defect, and as a result, they cannot make glucose when they're sleeping, and as a result, they could die under that circumstance. I'm not trying to bore you with this information. You must understand how these pathways are interrelated. Fatty acid oxidation. So with fatty acids, it's not the carbon that's used to make glucose. It's the energy of the fatty acid powering the synthesis of glucose. All right, now coming back to the normal situation here. So again, notice how the ATP coming from fatty acids are powering gluconeogenesis. At the same time, they're turning off glycolysis. So they don't work at the same time. And remember, when triglycerides are being broken down, they generate glycerol. And the measly 3 carbons of the glycerol from the triglyceride mobilization is not being wasted. It's now being used, saved, and used as carbon to make glucose. Now let me ask you this. We haven't talked about it. We will tomorrow. What's the end product of beta oxidation besides the ATP? What's the other end product of cycles of beta oxidation? What is the end product of beta oxidation? Not ketones, it is acetyl-CoA. And in the liver, you can make ketone bodies from acetyl-CoA, but it's acetyl-CoA. Does acetyl-CoA have anything to do with this figure here? Does acetyl-CoA have anything to do with page 206? That's right. There it is. Here it is. How the acetyl-CoA, the product of beta oxidation of fatty acids, is turning on, I should say, turning on gluconeogenesis. How one pathway turns on another pathway. Beautiful how this works. Beautiful. Now, I know what you're thinking. Ah, man, that's some biochemist up there getting off on all this biochemistry stuff. But if you think about it, you would say to yourself, man, my body is beautiful how it works. How these pathways are interrelated and they're interacting with each other. Turn on one pathway, you turn on another. Incredibly beautiful. All right, now to finish this off before the break. Now the fructose 6, look at all the color here. Look at all this color on this figure. We're still not done. All right, now fructose 6 converts to glucose 6-phosphate, and then the von Gurke's enzyme, glucose 6-phosphatase, then pops the phosphate group off to enable the glucose to exit the liver to maintain blood glucose values. All right, now one very last thing before the break. Integration with pharmacology here. All right, so you understand, look at all the color on this figure. All right, here comes the pharmacology. In pharmacology, what's the name of that well-known drug that's used to treat type 2 diabetes? Now, type 2 diabetes you can treat it with many kind of things like thiazolidinediones and sulfonylurea and so on. But what's the most common drug used to treat type 2 diabetes? That's right, metformin. Metformin. But now why? What's the biochemical explanation? Why do you give metformin? And the reason why you give metformin is to inhibit gluconeogenesis here. But now why do you want to do that? And here comes the answer. Because in a diabetic situation, there's plenty of glucose in the blood. Remember, in type 2 diabetes, the patient is hyperglycemic, usually due to the obesity business. But the idea is they are hyperglycemic. And because there's a lot of glucose in the blood, the body should be secreting insulin to take care of that hyperglycemia. But in type 2 diabetes, they don't have the insulin effect. Therefore, the body thinks you're starving. And when the body thinks you're starving, even though there's plenty of glucose in the blood, the body thinks you're starving. And when it thinks it's starving, how does the body respond? And on the left side of the page, it sends out hormones like glucagon, like cortisol. And what are they going to do? They're going to turn on gluconeogenesis to make more glucose, put it out in the blood, And that's the last thing you want to happen in type 2 diabetes, is to make more glucose when there's plenty of glucose in the blood. So therefore, you inhibit gluconeogenesis, and because you prevent the glucose from being made, exacerbating the problem. Now, the idea, so you understand that that's why you give metformin. And let me ask you this. In first aid, they talk about this as well. In pharmacology, what is the side effect, the main side effect of metformin administration? What's the main side effect? That's right, they have lactic acidosis. How do you explain that? How do you explain lactic acidosis when you treat with metformin? And here comes the answer. Because the target of metformin, the biochemical target, is pyruvate carboxylase. So you inhibit that enzyme by metformin, you don't make glucose. But the important feature is, by inhibiting that enzyme, notice its substrate is pyruvate. And I keep telling you, when you inhibit an arrow in biochemistry, the substrate accumulates. And when the substrate accumulates, it backs up. Pyruvate then backs up to lactic acid, which gets spilled in the blood, lactic acidosis in metformin administration. Everybody with me? Everybody, here it comes, get ready. It makes sense. There it is. A little bit of biochemistry goes a long way. All right, now we're going to take a 10-minute break and then come back, and a couple more pathways coming up. We're going to do alcohol metabolism coming up next, and then the all-important hexose monophosphate shunt in the next hour. All right, let's take a 10-minute break. Okay, see you shortly. Hello, everybody. Welcome back for the third hour. And where we're at is we just finished gluconeogenesis. And before we get to the last of the carbohydrate pathways, there's another pathway that we need to review because it's so high yield, and that's alcohol metabolism. Now, in terms of the biochemistry, it's very easy, but you're going to see why it's so important for board exams. Now, in terms of the biochemistry of alcohol, what happens is there's only one organ in the body that can metabolize alcohol, and that's the liver only. And the liver has two enzymes. Both of them will be highlighted in the red box. The first of these enzymes is called alcohol dehydrogenase. Look at the surname located in the cytoplasm of the liver. The surname is dehydrogenase, meaning it's going to pluck out some high energy electrons from carbon 1 of the ethanol, puts them on the NAD to form NADH, which goes into the electron transport chain, generating ATP. And then the product is called acetaldehyde. And then the enzyme acetaldehyde dehydrogenase, and there are two of these. One's located in the cytoplasm, the other is located in the mitochondria of the liver. And what they do is they take the acetaldehyde, more oxidation by removing electrons from carbon 1 aldehyde, makes it into a carboxyl group, but puts the electrons on the NAD to form NADH. And then the acetate product then goes into the Krebs cycle to be burned to make a lot more ATP. There it is. That's the biochemistry of alcohol metabolism. Now, what do you need to know for on board exams? Now, it turns out this is a relatively slow pathway. And so if somebody drinks too much alcohol, then what happens is alcohol as well as acetaldehyde, both of them can leak out of the liver. They can go into the blood and they can go to the brain. And when they go to the brain, they can impair brain function. And in fact, it's the acetaldehyde that can cause the brain hangover when somebody drinks too much alcohol. It's simply the acetaldehyde is the culprit in impairing brain function here and giving the hangover feeling when somebody drinks too much alcohol. Okay, now another item here is both of these enzymes are the targets in pharmacology. Why the pathway is so high yield for board exams? And another reason is because of the issue of pharmacology. Here it comes. In pharmacology, what is the name of that drug that's used to inhibit the alcohol dehydrogenase? The name of the drug used to inhibit alcohol dehydrogenase. Not disulfiram, Leo and Gisela, not disulfiram, Andrea, no. It is fameprazole, like Daniel mentioned. It's fameprazole, and so fameprazole inhibits alcohol dehydrogenase. Now let me ask you this. For what reason in pharmacology do you administer fameprazole? To treat what? To treat what? To stop them from drinking now? That's right, and the answer is for methanol poisoning or ethylene glycol. Methanol is wood alcohol. Ethylene glycol is the stuff that's in antifreeze. But the important feature is that both methanol as well as ethylene glycol, when they are ingested, they are seen by the same enzyme, alcohol dehydrogenase, and it oxidizes methanol to formaldehyde, and formaldehyde can leave the liver, get into the bloodstream, and formaldehyde it is the dangerous substance to the brain. And so, what fameprazole does, it inhibits the alcohol dehydrogenase from metabolizing methanol and ethylene glycol. And as a result, if they cannot be metabolized, then you'll pee methanol and ethylene glycol into the urine. They cause no problem unless they get oxidized to the aldehyde group. That's why you give fameprazole. Now, the acetaldehyde dehydrogenase, the second enzyme here, why is it important? Well, one reason why it's important is because the issue in many Asians, especially those in the Pacific Rim, like in the Philippines, in China, and in Japan, for example, it is the mitochondrial enzyme that's deficient. They still have the cytoplasmic enzyme, but the mitochondrial enzyme is deficient. And as a result, they cannot oxidize acetaldehyde to acetate very easily. And so acetaldehyde will accumulate in these individuals, and as a result, can go to the brain, and these people become alcohol intolerant. So if they ask you why, you say because it's the mitochondrial version of the acetaldehyde dehydrogenase that's deficient in many Asians. Not all Asians, obviously, but many Asians who are alcohol intolerant. But now here comes the pharmacology. Now in pharmacology, what's the name of the drug commonly used to inhibit acetaldehyde dehydrogenase? Now there you go. That's right. That's the disulfiram, also called antabuse commercially. So why do you give that drug in pharmacology? And the reason is you give it to people who are long-term alcoholics. Because by giving this drug to inhibit the acetaldehyde dehydrogenase, if an alcoholic were to drink alcohol, it would convert by the first enzyme to acetaldehyde, no problem. But if the second enzyme is being inhibited by disulfiram, acetaldehyde will accumulate. It'll spill out to the liver, into the blood, go to the brain, impair brain function, and those people therefore would then have a very sick feeling, a hangover feeling. They will throw up, and then psychologically, they will not want to drink alcohol. So it's a psychological situation where by inhibiting the second enzyme in biochemistry, then in behavioral science, you're causing these people to become sick if they drink alcohol, and therefore psychologically they will not want to drink the alcohol. There it is. That's the biochemistry behind the behavioral science, behind the pharmacology here. Okay, now another item here, a little oral vignette. Suppose they say to you that suppose you go out and run a marathon, 26 miles. So you go out and run a marathon, and after you run that marathon, obviously you're going to be very tired and you're going to be very thirsty. So you go to the local bar and you drink, and you order and drink and consume a 6-pack of beer. Shortly thereafter, you keel over and die. Here comes the national board with the question. What did you die of? In other words, here it is. Why is it bad to do severe exercise and then drink alcohol? What would you die of? Not dehydration, not hypoglycemia. Which acidosis, Andrea? There are many different kinds of acidosis. That's right, it is lactic acidosis. Lactic acidosis. How do you explain that? How do you explain you would die of lactic acidosis? This is how the national board asks you about metabolism without asking you what follows 1,3-BPG in glycolysis. They're not going to ask it that way. This is the way they do it, the integration of the pathways. So here's the answer. So when somebody's exercising, severe exercise, running a marathon, as we talked about the other day, because no human being can breathe in enough oxygen, that means there's going to be a lot of lactic acid being generated in the muscle, anaerobic glycolysis going on. And then the lactic acid leaves the muscle, and then in the muscle you get the cramping and the muscle weakness, and then the lactic acid will be dumped into the bloodstream. Then you get then you start slowing down when you exercise. But then the organ that's supposed to metabolize the lactic acid is the liver. So the lactic acid now comes into the liver. And in the liver, the enzyme lactate dehydrogenase, LDH, is supposed to take the lactic acid, uses NAD to convert to pyruvate. And pyruvate goes on to make glucose here. So again, this is all the LDH enzyme. Lactate dehydrogenase takes lactate plus NAD and then removes the electrons to form pyruvate. But the important feature here is, remember I mentioned to you, the amount of NAD in a cell is very tiny. But now if somebody drinks alcohol, all this lactic acid after they finish exercising, all this lactic acid in the blood heading over to the liver. But now in the liver, if somebody's drinking alcohol, notice alcohol metabolism requires NAD. Lactate metabolism requires NAD. And so that little bit of NAD around is now being diverted to alcohol metabolism. And then that lactic acid will start to accumulate in the liver, back up into the bloodstream, lower the pH, and you could die of severe lactic acidosis. There it is. There it is. It makes sense. There it is. Right there. As long as you know a little bit about chemistry, it goes a long way. Not hard. This is not hard. Understand why you would end up with lactic acidosis and die. There it is right there. As long as you know, the key here was understanding just a little bit of NAD in the cells. And now it's being diverted to alcohol metabolism. Okay, now what's coming up next is the last carbohydrate pathway to talk about. And this pathway is the all-important hexose monophosphate shunt. Now, also called the pentose phosphate pathway. But now, this is a pathway that if you were studying on your own, and you go to the Kaplan book or First Aid or some other book, and you came to the hexose monophosphate shunt, and you see all these arrows going up and down and all around, and you see these weird names of sugars and things like that, you might have a tendency to say to yourself, you know, maybe I'll take a chance that this pathway won't be on the national board. If you did that, you are hurting yourself. This is one of the most important pathways for the boards of them all, is the hexose monophosphate shunt. You just wait till we get into this pathway. It integrates with every discipline, is the hexose monophosphate shunt. It is such a high yield pathway. But you have nothing to worry about because I got your back. As long as you're paying attention, as long as you're following me, you will know what you need to know, and there's so much you don't need to know that I won't cover here. All right, now in terms of the biochemistry, here, let me do the biochemistry first. Now, once glucose enters the cell, it gets phosphorylated to glucose 6-phosphate. And then the glucose 6-phosphate has 3 pathways it can go into. One pathway shown here vertically is glycolysis. The second pathway shown on the right is the hexose monophosphate shunt. And then not shown on this figure, what other pathway can glucose 6-phosphate go into? Let me see your answer. What other pathway can glucose 6-phosphate go into other than glycolysis and hexose monophosphate shunt? What other pathway? Glycolysis is one, and the other is, that's right, I see it, and the answer is glycogen metabolism, glycogen synthesis. All right, so now we have to ask, why does the human body have 3 different pathways that glucose can go into? Glycolysis, you say, why do you have glycolysis? You say, well, that's pretty easy. The purpose there is to generate energy in the form of ATP. Why do you have glycogen? That's for purposes of storage, obviously. But now we'll address, why do you have the pathway of the hexose monophosphate shunt? It's not to generate ATP. It's not for storage purposes. So obviously we got to ask, why do you have the pathway of the hexose monophosphate shunt? We'll get into that in just a minute. But once it enters the hexose monophosphate shunt, right at the beginning, there is a major enzyme. Get that highlighter in all kind of colors, man. You got to know this enzyme, one of the most important enzymes of them all for the board exams. And that's glucose 6-phosphate dehydrogenase. Now, we'll talk about why it's so important on the next figure. But what does the enzyme do? Because it's a dehydrogenase, it means it plucks out high energy electrons from what? Glucose 6-phosphate, and puts those electrons onto NADP, not NAD. Puts them on the NADP to form NADPH as the product here. NADPH as the product, not NADH. Now remember, NADH is being used in the electron transport chain to generate ATP, We talked about that earlier tonight. But so then we got to ask, well, what's the function of NADPH? Remember, it's a reductant. It's a reducing agent. It's an electron donor. So as we'll talk about later, that NADPH is being used to take the electron and put it back into carbon, things like that. It's being used as a reductant, reducing agent. All right, now we'll come back to this enzyme a little bit later. Let's proceed. So then it makes a sugar product you don't have to know. And then there's another dehydrogenase that generates additional NADPH, additional reductant. And then it makes ribulose 5-phosphate, no big deal, which converts to ribose 5-phosphate, which is then used to make nucleotides to make DNA and RNA. So here comes the answer. What's the function of the hexose monophosphate shunt in case they ask you? You say there's two functions. One is to generate reducing power in the form of NADPH. Secondly, the function of the pathway is to make ribose, to make nucleotides, to make DNA and RNA. That's the function of the pathway here. Now the ribose 5-phosphate, if it's not used to make nucleotides, then the ribose can go into another part of the pathway, which is absolutely useless information for your purpose, low yield. And it makes these weird sugars. Don't worry about the sugars, weird sugars. And some of these sugars in the brain are needed for proper brain function here. And so, these make these weird sugars. Don't worry about them at all. So the rest of the pathway is absolutely useless, with one notable exception. In the other part of the pathway, there is a major enzyme you must know, and that enzyme is called transketolase. What the substrate of the enzyme is, what the products, the sugar products of the enzyme are not important. Don't worry about what the enzyme catalyzes, not important. But what is important, it needs thymine pyrophosphate. You must know it requires thymine pyrophosphate to take the sugar substrates to make other sugar products. And in the brain, these sugar products are necessary for proper brain function here. So what I want to do right here is right at the beginning now, start talking about some high yield items. And the first thing I want to talk about is the list, the thymine pyrophosphate requiring enzymes. And there are 4 of them. Now the first of the thymine pyrophosphate requiring enzymes obviously is transketolase. Now what's another thymine pyrophosphate requiring enzyme that we talked about earlier? What's another one that we talked about? Give everybody a chance. That's right. Ifosa, that's right. And that is PDH, pyruvate dehydrogenase, that we talked about yesterday. Now there was one that we skipped that was in the Krebs cycle, and that's alpha ketoglutarate dehydrogenase. That's a third one. That's a major enzyme of the Krebs cycle. And then the fourth one, the last one, that's the key enzyme that's defective in the disease maple syrup urine disease. Anybody know the name of that one? Now be careful how you pronounce it. Here. It's not branched amino acid. No, it's not branched AA. No, it's branched chain keto acid dehydrogenase, not amino acid dehydrogenase. I'll explain why on Friday. But the name of the enzyme that's when we'll talk about the enzyme. It's the branched chain keto acid dehydrogenase here. Now, of the 4 enzymes that need thymine pyrophosphate to work, notice it's the bottom 3 that are the 3 tender loving care for Nancy enzymes that we talked about late yesterday. PDH, alpha KG dehydrogenase, and branched chain keto acid dehydrogenase. Notice that these are dehydrogenases. All 3 of them make coenzyme A of products and so on. Transketolase is not. Transketolase only requires thymine pyrophosphate to work, but doesn't make coenzyme A products, doesn't do any other thing the other enzymes do. So now, in this yellow box, this alone would say why this pathway is so important for the boards. But this is just the start. All right, now coming up next here is pathology. Now, please pay attention, because I'm telling you right now, if you're not paying attention, if you're being diverted by something else or distracted with something else, or you're watching a basketball game on TV or something like that, I'm telling you right now, that you'll come back to this in a few minutes, and then you'll start saying, I don't understand what you said. Or you're going to go to the Q&A pod, and Dr. Saida, Dr. Aaron will have to answer. And now let me stress here, I don't mind repeating the explanation. But as long as you're paying attention, if you still don't get it, I'll explain it again. But if you're not paying attention, I guarantee you're going to start asking questions. I don't understand what went on if you're not paying attention, because the next part is somewhat tricky. How do I know that? History tells me so. Every time I talk about this next part, many students will come up to me at the break when I do live lectures, for example. I don't understand what you said. Well, and then I ask, were you paying attention? Or were you texting somebody on the phone? And then because you're not going to understand it. So please pay attention to this next part here. All right, here it comes. Now what happens is there is a genetic disease out there. And this genetic disease is called Wernicke-Korsakoff disease. It's a genetic disease in which the gene for transketolase is genetically defective. It has a missense mutation. So because of the missense mutation, the missense mutation is in the gene, the enzyme transketolase is made, but the defect is in the active site, and it doesn't bind thymine pyrophosphate very well. It does not bind thymine pyrophosphate very well, and so therefore the enzyme won't work. And when the enzyme doesn't work, the enzyme doesn't make those sugar products, and therefore the brain doesn't work, and you end up with Wernicke-Korsakoff syndrome. Now Wernicke-Korsakoff syndrome is the pathology. It's the clinical manifestation when this single enzyme doesn't work. So one more time. Wernicke-Korsakoff disease is a genetic disease in which the patients have a missense mutation in the gene for transketolase. The enzyme is made at the gene level, but because of the mutation, it doesn't bind thymine pyrophosphate very well, and therefore it doesn't work very well. It doesn't make the sugar products, and as a result, the brain is impaired. And then the pathology is Wernicke-Korsakoff syndrome. The syndrome is the pathology when this enzyme doesn't work. Okay, everybody with me so far? Yes or no? Everybody with me? Everybody with me? All right, so far, so good. All right. Because a lot of pathologists get this wrong. A lot of pathology people, they misinterpret what's really going on biochemically. All right, so what happens is somebody came up with a bright idea. Why not give these patients who have a genetic disorder in transketolase, why not give them extra thymine? Because if you give them extra thymine, then it forms thymine pyrophosphate. It forces itself into the defective active site of the enzyme, and now the enzyme will start to work better. It makes those sugar products, and then Wernicke-Korsakoff syndrome is overcome. Even though they have a genetic disease, Wernicke-Korsakoff syndrome, the pathology, is overcome by giving them thymine supplements. So this is a genetic disorder helped nutritionally. By giving them thymine, it forces itself into the defective active site of the enzyme, and the enzyme now works better, and the pathology is overcome. Everybody with me so far? Everybody with me? Don't worry about alcoholism, Eamon. We're not talking about alcohol. Just focus on what I'm talking about here. Don't worry about anything else. We'll talk about alcoholism a little bit later. Focus on what we're talking about. You'll never understand alcoholism unless you understand this. Okay, everybody with me? Okay, all right. Okay, so then what happened is, so again, this is genetic disorder helped nutritionally. Okay, so then what happens is as time goes on, people start to notice that these people who had Wernicke-Korsakoff syndrome, the pathology because of the genetic disorder, taking pills of thiamine, thinking, oh, I know why they're taking thiamine supplements, because they're thiamine deficient. They're not thiamine deficiency. They're not thiamine deficient. What's thiamine deficiency called? Let me see your answer here. What is thiamine deficiency called? What's it called? What's thiamine deficiency called? That's right. It's called beriberi. All right, so now we're going to try to explain what's the difference between Wernicke-Korsakoff syndrome and beriberi. They are not the same. And I'm telling you right now, a lot of pathologists get this wrong. So again, they're not the same. Now remember, transketolase is the enzyme responsible for Wernicke-Korsakoff syndrome. The pathology, the ataxia, the psychosis, the confusion, the ophthalmaloplasia, the wide-eyed appearance, that is because of transketolase not working. And transketolase might not work for two reasons. One reason shown in the purple box is because of genetics. One enzyme not working, you end up with Wernicke-Korsakoff syndrome. Now remember PDH, that's the enzyme responsible for lactic acidosis. When that enzyme doesn't work, then pyruvate accumulates and spills into lactic acid here. All right, so now, what is beriberi in thiamine deficiency? Now notice what's going to happen in beriberi, thiamine deficiency. The idea there is if someone's thiamine deficient, transketolase starts not to work. The brain now becomes impaired with Wernicke-Korsakoff syndrome. But now that's complicated because now PDH starts not to work, giving rise to lactic acid. And lactic acid can also get into the brain and impairing brain function. And that is dry beriberi. So dry beriberi, the neurological problems, is a combination of Wernicke-Korsakoff syndrome because transketolase is not working, and also because of the lactic acidosis that goes on here. And that's dry beriberi. Now, as time goes on in thiamine deficiency, what's going to happen is several other enzymes in metabolism start not to work. Alpha-ketoglutarate dehydrogenase, one of the top enzymes of the Krebs cycle. Branch-chain ketoacid dehydrogenase, one of the top enzymes of amino acid metabolism. And remember, including PDH, these are ATP generating pathways. So you can imagine now, as time goes on, when someone is thiamine deficient, major enzymes in ATP production pathways will not work, not enough ATP is being made, and so what happens? Then the heart starts to pump harder and harder. Why? To start getting, its body thinks you're starving for fuel, so the heart starts to pump harder to try to generate fuel to burn to make ATP. But not enough ATP is being generated. The heart's pumping harder and harder, high cardiac output to try to get the carbon fuel to burn to make ATP. High cardiac output. Now that's wet beriberi. And as time goes on, if someone's thiamine deficient, not enough ATP, ultimately cardiac failure. There it is. There it is. That explains the wet beriberi. So again, one more time. It starts off, if someone's thiamine deficient, it starts off as dry beriberi, which is a combination of Wernicke-Korsakoff syndrome, the ataxia, confusion, and the lactic acidosis. And as time goes on of thiamine deficiency, it manifests to wet beriberi, starts to involve the cardiac system to try to pump to generate more ATP. Eventually, cardiac failure will occur. Now, Iman, you ask about alcoholism. So why is it alcoholics present with Wernicke-Korsakoff syndrome? And its answer is simple, as long as you were paying attention. The answer is simple. Is alcoholics, remember, are thiamine deficient because alcohol inhibits thiamine absorption. and there's no thymine in alcoholic beverages. So alcoholics tend to be thymine deficient, therefore transketolase will not work, and as a result, they're going to end up with the Wernicke-Korsakoff syndrome, compounded now because lactic acid will start to accumulate. There it is. There it is. There it is. As long as you're paying attention, you now know the difference between Wernicke-Korsakoff. You now know wet, dry, berry. You now know why alcoholics present with Wernicke-Korsakoff. You must understand. You can't memorize this stuff. You can't ask for mnemonics here. You can't memorize. You must understand. And as a physician, you should want to understand this information because it's applicable to your profession. You are going to see alcoholics with the Wernicke-Korsakoff. The sooner you understand, the better. Now, if you think transketolase is high yield, and it is, you wait till we get to G6PD. It is even more important for the National Board. All right, let's talk about this enzyme, G6PD. Now, to understand what goes on with that enzyme, we must understand what's the function of NADPH. And what's the function of NADPH in a cell? Depends on three things. It depends on location. What's the other one again? I forgot. Location. Oh, yeah. Location. It depends on those three things to understand what's the function of NADPH. Now, what I'm going to do right here is I'm going to take this part of the pathway right here, this part of the pathway right there, and move it into different locations and ask, what's the function of NADPH in these various locations? Now, that's shown. I'm going to take this part of the pathway and now first move it into the liver. This is now on page 211. Now you see page 211 in front of you there in your book. You might want to put a big bookmark on this page. It is the day before you take the National Board. You might want to revisit this page. It is so high yield. Every discipline is on page 211. Every discipline. All right, so now we're going to take this part of the figure right here. Here's that part of the figure. Here's the G6PD, here's pentose phosphates, and here is the NADPH. Now in the liver, what's the function of the NADPH electron? Well, in the liver, it's being used for biosynthetic reactions to put the electron back into carbon, to make fatty acids, to make cholesterol, to make nucleotides, things we'll be talking about over the next couple of days here. So in the liver, it's being used for biosynthetic reactions. Now we're going to take this part of the figure right here and move it into another cell type in immunology, and that is the neutrophil that Dr. Moscatello talked about. Here is the enzyme G6PD, and here is the NADPH product. Now in the neutrophil, or any phagocytic cell, what's the function of the electron in NADPH? Well, as you all know, in immunology, it's being used on purpose to put the electron on to oxygen to make superoxide by that all-important enzyme in immunology called NADPH oxidase. So you put the electron on the oxygen to make superoxide, and then it converts to hydrogen peroxide by other enzymes, and then it's being used as part of the respiratory burst, along with hypochlorite and so on, to kill microbes. The normal biochemistry related to the immunology. But here comes the pathology. Now in pathology, in G6PD deficiency, to understand what goes on, all you got to do is draw arrows up or down. In G6PD deficiency, the arrow is down in the neutrophil. What about the ability to make NADPH in the neutrophil? The answer is it's down. What about, therefore, the ability to make superoxide? Down. What about the ability to make hydrogen peroxide? Down. What about the ability to kill microbes? Down. So you would imagine, therefore, in immunology and in pathology, one of the hallmarks of G6PD deficiency must be and is immunodeficiency. These people are very susceptible to infections. Now, the national board is going to love this. Why? There's another disease out there that's very similar to G6PD deficiency, in which those patients are always susceptible to infections. And now, what's the name of that disease? I just looked over and I saw it. That's right. What's the name of that disease? That's right. Well done, everybody. And that is chronic granulomatous disease. Now, chronic granulomatous disease, what is the biochemical defect? And the answer is, in chronic granulomatous disease, it's where the NADPH oxidase enzyme is genetically defective. The gene is mutated, and so the enzyme is defective. So there's plenty of substrate, NADPH, but the enzyme is defective. Therefore, they can't make superoxide, hydrogen peroxide. They can't kill microbes, and therefore they're very susceptible to infections. So the National Board loves this. Why? Because they'll talk about a patient in the first sentence or two. The patient is very susceptible to infections and blah, 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 and they're going to want to know, how do you know? Is it due to chronic granulomatous disease, or is it due to G6PD deficiency? So here comes the question. How are those two diseases distinguished from each other? How are they distinguished from each other? And the answer is, that's right, it's the other two features of G6PD deficiency. And what are they? What's the other two main features of G6PD deficiency? And one of them is, that's right, it is the Heinz bodies. And other people, including Mylan and so on, said hemolytic anemia. Here, so again, characteristic of G6PD deficiency. Let me ask you this. In chronic granulomatous disease, yes or no? Do they have Heinz bodies? Yes or no? In chronic granulomatous disease, do they have Heinz bodies? The answer is no. In chronic granulomatous disease, do they have hemolytic anemia? Yes or no? Do they have hemolytic anemia in chronic granulomatous disease? The answer is no. Why not? Because of three things. Location, location, location. In chronic granulomatous disease, there's nothing wrong with the erythrocyte. The erythrocyte is fine. And Heinz bodies and hemolytic anemia have to do with the erythrocyte. And in chronic granulomatous disease, nothing wrong with the erythrocyte. That's why they only have the immunodeficiency in chronic granulomatous disease. Now, coming back to G6PD deficiency now, we got to understand what's going on in the erythrocyte with the NADPH electron. So what we're going to do now, I'm going to take this part of the figure right here, this part of the figure, and now move it into the erythrocyte and ask, what's the function of the NADPH electron? So here it is. Here is the G6PD. Here is the NADPH. Now, as we all know, what's the function of the NADPH electron in the erythrocyte? Unlike the liver, it's not being used to purposely put it back in the carbon to synthesize things. The erythrocyte synthesizes nothing. Unlike the neutrophil, it's not being used to purposely make superoxide. So what's the function of NADPH electron in the erythrocyte? Well, as everybody knows, it's being used for detoxification capacity. All right, now, so the idea is now in the erythrocyte, there are a lot of free radicals being generated, as we talked about yesterday. Now, why are so many free radicals being generated in the erythrocyte? As I mentioned yesterday, some of the oxygen, when it goes through the bilayer, can spontaneously pick up an electron from the carbon-carbon double bonds. Remember all that of the fatty acids of the phospholipids, and thereby spontaneously make free radicals. But most of the free radicals in the erythrocytes come another way that I did not talk about. I'm going to talk about it now. And here it is. In the erythrocyte, suppose this represents hemoglobin. One of the 4 subunits of hemoglobin, and in parentheses is one of the 4 heme pockets of hemoglobin. And in the heme pocket, there's iron in the +2 state in the heme. And then now when you breathe in the oxygen goes through the bilayer, and oxygen now will go on to the heme pocket of hemoglobin. Now, then the red blood cells will leave the lung area. As they travel through the arteries, what can happen is the oxygen can take the electron as it sits there in the heme pocket. Oxygen can take the electron from the iron in the +2 state. And by doing so, by accident, it takes the electron. Remember, oxygen is an oxidant looking for electrons. By accident, oxygen can take the electron from iron +2 up to about 3% every day, and thereby makes two products. One of the products is superoxide by taking the electron. The other is methemoglobin. Remember I mentioned that yesterday, that you and I generate methemoglobin every day. And this is how you generate it, by accident. Now, of these two products, if that's all there was, after a week to 10 days, you'd be dead. All your hemoglobin would be methemoglobin, you couldn't carry oxygen, all the superoxide, destruction, you'd be dead in no time. The body says, you know, I want to live. And if you want to live, you got to do something about this. How does the body deal with these two products? Well, how does it deal with methemoglobin? And the answer is there is an enzyme, low yield, but it's called methemoglobin reductase, that takes the electron from NADH in glycolysis and puts the electron back into iron +3 to restore hemoglobin, iron +2. Now, as we talked about yesterday, this methemoglobin reductase enzyme can be genetically defective. And as a result, that gives rise to the disease and pathology called methemoglobinemia, where the methemoglobin levels are about 30% compared to 3%, and these people look blue. They have a tinge of blue to them, cyanotic. All right, now here comes the question. In pharmacology, what is the name of the drug used to treat methemoglobinemia? What's the name of the drug used to treat methemoglobinemia? And man, Bobby, where'd that come from, this statement? Okay, you must have been ready for me. You write very fast. But the answer is, it is methylene blue is the primary. You could use vitamin C, but the main one is methylene blue. But now, why do you give methylene blue? That's right, to ask you to study. Why do you give methylene blue? Bobby was cooking his answer. Why do you give methylene blue? Because methylene blue is a reductant. It's a reducing agent. It's an electron donor. So by giving methylene blue, it's substituting for the enzyme. The enzyme is defective. And now methylene blue chemically will give up its electron to iron +3 to restore iron +2. That's why you give methylene blue. It's a reductant. Now remember, methylene blue is the opposite of nitrite. Remember earlier this evening, we talked about nitrite in the treatment of cyanide poisoning, because nitrite is an oxidizing agent. You're purposely making methemoglobin. But here, methylene blue is the reverse. It's a reductant. It's giving electrons to restore hemoglobin from methemoglobin. All right, so there it is. Now, how does the body deal with the superoxide? So we'll finish this figure, then we'll take the 10-minute break. How does the body deal with the superoxide? Well, there's an enzyme that will convert the superoxide to hydrogen peroxide. And the name of that enzyme is called, superoxide dismutase. So it makes hydrogen peroxide. And hydrogen peroxide is looking for 2 more electrons to form water. And where are those 2 electrons coming from? And where they're coming from is, they're coming from a substance called glutathione, reduced glutathione. You need 2 molecules, that's what it stands for, 2. G stands for glutathione. And what's glutathione? It's a tripeptide, 3 amino acids. Now the 3 amino acids you don't have to know, except the one in the middle. The amino acid in the middle is called cysteine. And the SH represents the sulfhydryl group of cysteine of glutathione. Remember, we talked about cysteine way back on page 119. But that is the sulfhydryl group of cysteine of glutathione. Remember, sulfhydryl groups are electron donors. I mentioned that earlier. Because it's a reductant. What happens is there is a low yield enzyme called glutathione peroxidase, which requires selenium. And what does the enzyme do? It takes 2 electrons from 2 molecules of glutathione, takes the sulfhydryl group electrons, gives them to the hydrogen peroxide to form water. It's now detoxified. But now what happens to the sulfurs? When the sulfhydryl groups give up their electrons to detoxify, the sulfurs then cross-link. When they cross-link, they generate oxidized glutathione. Now, if that's all there was, then after a short period of time, all of the glutathione would be oxidized and you couldn't detoxify anymore. So you would imagine some way, somehow, we got to take oxidized glutathione and restore reduced glutathione to keep the detoxification going. And so where are those electrons coming from to restore oxidized glutathione back to the reduced glutathione? Well, sure enough, it's coming from the NADPH, which comes from the enzyme G6PD, which comes from the glucose that you eat. The NADPH electron sees a low yield enzyme, another one called glutathione peroxidase, reductase I should say. It takes electrons from NADPH, gives them to the oxidized glutathione to restore reduced glutathione to keep the detoxification going. There it is. That's what's the function of NADPH in the erythrocyte. All right, now, in pathology, in G6PD deficiency, what happens is all you got to do is draw arrows up or down in the erythrocyte. In the erythrocyte, what about the ability to make NADPH? The answer is because the enzyme in G6PD deficiency is down, then the ability to make NADPH is also down. What about the ability to make reduced glutathione? Down. What about hydrogen peroxide, therefore? It starts to rise. And when it starts to rise, here comes the Heinz bodies, and here comes the hemolytic anemia. All right, we got explaining to do. How do you explain the Heinz bodies? Here are Heinz bodies. Now remember from pathology, these are erythrocytes, and in the middle of the erythrocytes are these solid black spots. And these are Heinz bodies. And notice how some of the Heinz bodies are right there in the cytoplasm, and many Heinz bodies are attached to the membrane, like here and here and here. So how does biochemistry explain the Heinz bodies? The answer is simple. In the erythrocyte, here is hemoglobin. Now remember, hemoglobin has 2 alpha and 2 beta subunits. And on the periphery of each of the subunits, there are cysteine residues. And these SHs here represents the sulfhydryl groups of cysteine on the periphery of hemoglobin. All normal. But when hydrogen peroxide starts to accumulate, hydrogen peroxide is an oxidant, sulfhydryl groups are reductants. What do you think is going to happen? That's right. The hydrogen peroxide now will take the electrons from the sulfhydryl groups to detoxify to form water. But now what happens to the sulfurs of the hemoglobins? They start to cross-link with each other, forming disulfide bridges. And that is now a Heinz body. A Heinz body is covalently cross-linked, denatured, precipitated hemoglobin. When hemoglobin cross-linked with each other by these disulfide bonds, it precipitates and denatures. And that's what this depicts here, is hemoglobins cross-linking with each other. That's those little black spots called the Heinz body. And notice that some of these Heinz bodies can attach to the membrane. In the membrane, there are sulfur lipids. And sure enough, a piece of junk, a Heinz body, now attaches to the membrane. What do you think is going to happen to the membrane when a piece of junk is attached to the membrane? The membrane can rip. And when the membrane rips, it becomes leaky, and there's the hemolytic anemia. It's simply the membrane integrity is impaired. Not only that, hydrogen peroxide can punch holes in the membranes, things leak out, hemolytic anemia. it makes sense. All right, now we're still not done with this. Just another minute or two, then we'll take the break. But the idea of pharmacology. Now remember in pharmacology and pathology, G6PD is highly polymorphic. In other words, some people in this world have a mutation. Depending on the mutation, remember allelic heterogeneity in genetics? Allelic heterogeneity. We talked about that a few days ago. But depending on where the mutation is, some people have 90% of the activity compared to normal. Everything is fine. Other people have another mutation in the gene for G6PD. They may have 80% of the activity compared to the worldwide population. Everything is fine as well. Other people have 70%, 60%. And everything is okay until we get people in this world, depending on their mutation, they end up with about 50% of the enzyme activity compared to the worldwide population. And that's what's referred to in pathology as episodic G6PD deficiency. In other words, they have just enough enzyme activity to make just enough NADPH, to make just enough reduced glutathione to detoxify what hydrogen peroxide is normally being made. They're at the maximum capacity to detoxify. But if there's some episode, and here come the episodes, all those infections you saw on the previous page, the issue of the neutrophils and so on generating extra hydrogen peroxide, and here comes the pharmacology. You know, in pharmacology there are at least 50 drugs or more. Drugs like sulfa drugs and so on, you never administer to anybody with G6PD deficiency. And the reason for that is because those drugs generate extra hydrogen peroxide. And by generating extra hydrogen peroxide, you overwhelm the limited capacity to detoxify. You can end up with a hemolytic anemia episode. And fava beans. Now, fava beans, you may or may not know, most of you know this, but fava beans is a food staple in the Mediterranean countries. And what happens is fava beans generate a substance which generates extra hydrogen peroxide, overwhelming the limited capacity to detoxify. Matter of fact, a few years ago at my institution, when I was lecturing on this material, later on that afternoon, a medical student came up to my office and said years ago when he was a child, he was experiencing a bout of hemolytic anemia. So he said his parents rushed him to the doctor, and the doctor could not figure out what was the source of the hemolytic anemia. He couldn't figure out what was causing the hemolytic anemia. So he said that they airlifted him to the University of Cincinnati Children's Hospital. And even there, they couldn't figure out what was causing the hemolytic anemia. He said up there the doctors were doing blood transfusions on him and so on, and they couldn't figure it out until some doctor looked at his chart. And when the doctor looked at his chart, the doctor noticed that his name sounded foreign. It wasn't like Joe Smith. It sounded like it was a foreign sounding name. So he said the doctor asked his parents, excuse me, can you tell me what your nationality is? Where are you from? And he said, we're from Egypt. Ah, Egypt, Mediterranean country. What did you have for dinner last night? And sure enough, it was a fava bean food. So he told me, he said his parents almost killed him by giving him a fava bean food. Now, this is a true story, and why am I telling you this? Because it would be to your benefit. Before you take the National Board exam, you might want to get a geography book out there. And when you get the geography book, find out what countries are in the Mediterranean countries so that you know that Argentina is not a Mediterranean country. Because the way they're going to do this on a national board is they're going to say a 16-year-old Italian male, or a 20-year-old Greek male, or a 15-year-old Moroccan male, X-linked recessive business. And so you got to know this because they'll give you the clues in the first phrase there. Okay, now what we're going to do is we're going to take a 10-minute break and then come back, and then Carrie is going to set up the timer for 60 seconds after the break, and then there are going to be 3 questions coming your way, 3 vignettes, and she'll bring up the selection pod after the break. So let's take a 10-minute break right now, and I'll see you shortly, and then we'll do some vignettes. See you shortly. Okay, everybody, as promised, there's going to be 3 questions coming up here, vignettes based on stuff we've talked about thus far. And I'm hoping you get at least 2 of the 3, if not all 3 of them here for these 3 questions coming up here. Okay, so what we're going to do, I'm going to give you 1 minute, which is less than you get on board exams, but I'm just trying to speed you up a little bit. So then you make a selection, and then we'll close the poll, broadcast the results, and then do an explanation of each of the 3 questions. So here is the first question. All right, so 1 minute. Ten seconds. Okay, and end the poll, and looks like, and broadcast the results, and looks like, wow, so all over the place. 43% of students put option D was the majority, although a lot of people put option A, and the answer to this question is option D. Glycogenolysis. So the majority of students did get it correct. 43% of students is option D. All right, so what are we talking about in this vignette? What we're talking about is anaerobic exercise. Olympic runners, you hear about this all the time. And so they run, so those people like Usain Bolt, when they run a 100-meter dash, they accomplish that in less than 10 seconds. Olympic records like 9.5 seconds for 100 meters. All right, so the idea is, so this is all anaerobic. So the question is, where is the ATP coming from to do all that exercise in those 10 seconds? And now the idea is it's coming from glycogenolysis. Now, before we explain why that's the correct answer, let's do the other ones. The ATP stores. Now, ATP is gone in less than a second. You're not storing ATP like you store fat or whatever. So the ATP itself in the cell, any excess stuff is gone in less than a second. When you take a first jump or two, it's basically gone in terms of the storage, the excess ATP you might have around. Now, creatine phosphate, a lot of people put that. And that's actually gone in 4 seconds here, before the 10 seconds for the running of this dash. Now, what I want to do is a quick review. Now's the time to review creatine phosphate. Now, what creatine is, just to review this, no one expects you to know the structure. But sometimes if you see structure, you remember it a lot better. But this is creatine on the left. And then there's that all-important enzyme called creatine kinase. Now, as you know, there's several different isoforms of creatine kinase. There's the MM, MB, and BB isoforms. Remember, let me ask you this. Which creatine kinase is the isoform that's the one that's indicative of an MI? And if it's in the bloodstream, which creatine kinase isoform? That's right, it's the CKMB. That's right, the MB isoform is indicative of cardiac muscle damage. But in any case, these creatine kinases use the high energy phosphate of ATP, and they stick it onto the nitrogen. And by doing so, it makes creatine phosphate as the product. Notice that there's a direct linkage of phosphorus, not phosphate, phosphorus atom on nitrogen. That's what makes it high energy. And it's very unstable. A phosphorus atom on nitrogen atom. And because it's high energy, it can go in the reversible reaction. This is a highly reversible reaction. And because it's unstable, the phosphate can come off and thereby then the rest of the molecule cyclizes to make creatinine, and that's dead end. And it's spilled into the urine, and as you all know, that the amount per day is constant and depends on the muscle mass. And the creatinine is then used as a normalizing factor for things getting into the urine, used as a normalizing factor. Okay, so a quick review of creatine phosphate. Now, next one here is gluconeogenesis. Now, why is that incorrect answer? Because gluconeogenesis is being kicked in at the gene level, and it takes hours to kick in. So you're not getting ATP from glucose being made by gluconeogenesis because it takes hours. Now, the lipolysis that will eventually kick in. You secrete epinephrine. Epinephrine will go to the adipocytes to break down triglycerides to fatty acids, and that will be a major source aerobically after about 10 seconds. But so not in the 10 seconds, because fatty acid oxidation depends on oxygen. Now the last one here is option D. The correct answer is glycogenolysis. Now what happens there is in the first 10 seconds of running, because you're consuming ATP down to ADP, there is a low-yield enzyme that interconverts two molecules of ADP to make AMP and ATP. More ATPs being made to do muscle contraction, and then you generate AMP. And remember, the AMP is going to stimulate glycogen breakdown. Remember, the phosphorylase also stimulate glycolysis in hard-working muscle. That's where the AMP comes from in the first place. Also, ATP is being generated by the creatine phosphate business at the bottom of the figure. But again, that lasts about 4 seconds. And then you also, anaerobic glycolysis also generating a couple of ATPs. And where is that glucose coming from? To do anaerobic glycolysis, it's coming from glycogen breakdown. That's why the answer is D. Now, because in that 10 seconds, you're not breathing in very much oxygen, then all that lactic acid, the end product of anaerobic glycolysis, spills out of the muscle, gets into the blood. Therefore, you increase blood lactate levels, and lactic acid then lowers the pH of the blood from 7.4 to about 7.24, and that's why you start slowing down. There it is there. A quick review of anaerobic exercise here. All right, now what's coming up next is question number 2. Okay, I hope you get this one. It's not that hard in my opinion. I think most people get this next one. Here's question number 2. Ten seconds. All right, there it is, and 1 minute, and let's end the poll and broadcast the results. Okay, so looks like, I thought more people would have got this one. Looks like 40% of students put option B. Option C and D also are chosen, and the answer to this question is option B, complex 4 in the ETC, electron transport chain. What's the problem in this woman? What is her problem? The toxic effects of what? What's the problem? What's the problem in this individual? Let me see your answers. Not oligomycin. Good try, Mylin, but not. No, it is cyanide poisoning. Cyanide. Now, how do you know that? Because of this phrase. Odor of bitter almonds. That's a characteristic of cyanide poisoning, is the odor of bitter almonds. By the way, I've never done the experiment to see, do they have an odor? Does cyanide poisoning have an odor of bitter almonds? I don't want to do the experiment to test myself, but that's what they say. And so on board exams, if you see odor of bitter almonds, or they say there's a house fire, and furniture is burning, and sometimes furniture generates cyanide. If they say something about odor of bitter almonds, you automatically think they're talking about cyanide poisoning. And then in pathology, the cyanide here is the characteristics, the bilateral ptosis and symmetric weakness of the upper and lower extremities, all characteristics of cyanide poisoning in pathology. All right, so the question now becomes is not what is the name of the problem, cyanide poisoning. They're asking, well, what does it do? And what it does, it prevents, it inhibits complex 4 in the electron transport chain because cyanide binds the ferric iron in complex 4. All right, now let's go through the other 4 options here. What did we talk about that inhibits option A? What did we talk about that inhibits? We're talking about inhibitors now, these 5 items here. What did we talk about that inhibits option A? Adenyl cyclase. What does that? There is something that inhibits it. Not PDE. No, not PDE. What inhibits adenyl cyclase? Specifically, what inhibits adenyl cyclase? And the answer is, gonna move on, not methylxanthines, no, not exotoxin, no. And the answer is, it is alpha I of GI. Remember, the inhibitory G protein, GI, alpha I binds GTP, and alpha I inhibits adenyl cyclase. So we're talking about inhibitors of these 5 things. Cyanide inhibits complex 4. Now, what did we talk about earlier that inhibits option C, the phosphodiesterase in the liver? What did we talk about that does option C? Inhibits the phosphodiesterase in the liver. Now I need some of you earlier. That's right. There we go. That's right. That was the methylxanthines, and the key there was the liver, not sildenafil, because that's smooth muscle. The key here is the liver. Option D. What did we talk about that inhibits the phosphofructokinase 2? What did we do that does that? Option D. That's right. That's glucagon. That's right. Glucagon activates protein kinase A, which phosphorylates the PFK2, turning the enzyme on. Now, option E, we didn't talk about it directly, but you did in physiology, and you certainly will in physiology. What inhibits the sodium potassium ATPase? What does that? That's right. That's Walbane is an inhibitor. Also, digoxin does that as well. Inhibits the sodium potassium ATPase. So notice these are all inhibitors of the respective options here. Now the last question is a little bit harder one. You have to take. It takes a lot of thought. You can't memorize for this last question. You have to think overall what's happening. All right, here it is, the third and last question. Ten seconds. All right, and wow, I broadcast the results, and they're all over. They're all over. Wow. We're all over the place, and the answer to this question is, that's why I'm doing these questions. You know, if they were simple questions, everybody gets every simple question, then it doesn't do you any good. So we learn by these questions here. And the answer to this question is option E, translocation of GLUT4 to the cell membrane of the myocytes. Now, in this vignette, what we're talking about is exercise-induced hypoglycemia. Now, they're saying that this woman, she has type 1 diabetes, but she's controlling that. And she says after she had exercise, she became dizzy, nauseated, and feels faint. And they want to know what's the underlying mechanism of that episode, the episode of a brain problem. Dizzy, being nauseated, and feeling faint. Those are neurological problems. What's the basis of that? And the answer is option E, translocation of GLUT4. Remember what we talked about. I told you that when you exercise, you put GLUT4, again, this is diabetes. She's feeling dizzy, nauseated, and feeling faint. And so talking about a brain problem. The brain is getting dizzy here. But the idea is that when you exercise, then exercise causes GLUT4 to be put onto the surface of the muscle cells of the myocytes. And so now the muscle cells, because there's more GLUT4 on their surface, the muscle cells drawing glucose out of the blood to carry out muscle contraction, to burn that glucose, to make ATP, to do muscle contraction. So what that means is the body levels of glucose now start to go down. Why? Because the muscle is drawing the glucose out of the blood. And because the blood glucose is dropping, the brain is not getting its fair share. And when the brain doesn't get its fair share of the glucose, it starts to become dizzy, nauseated, and feeling faint. There it is. Typical board question. You have to think globally what's happening in the body. Now the other options here, option A, inadequate delivery of oxygen to the muscle. I mean, that would have producing pain and cramping, but the question is not asking about that. The question is not asking about the muscle. The question is asking about the brain. Why is the patient dizzy, nauseated, and feeling faint? That's a brain issue, not a muscle problem here. Now, option B and C, stimulation of gluconeogenesis in the liver and glycogenolysis in the liver. They would raise blood glucose. If they were stimulating glycogen breakdown, if anything, that would be helping the brain. So you can eliminate those. They would raise blood glucose, not lower blood glucose. Option D, stimulation of glycogenolysis in the muscle. Why is that incorrect response? Because the muscle has nothing to do with maintaining blood glucose here. The muscle has that glycogen, but it's a selfish organ. It keeps it for itself. So the only item here is what's causing the brain to not get its fair share of glucose. It's because more GLUT4 is on the surface here. There it is. It makes sense. This is a question you can't memorize. You can't, you have to think. You have to think. You got to use your beautiful mind. You know what's going to happen to all those medical students that are going through the motions of just memorizing things. If you try to memorize, you're not going to get this answer. You aren't going to get the answer if you're a memorizer. And if you're a memorizer, you're like all those other medical students. They're like zombies. They're going into the black pit of memorization. They are learning nothing. But so again, don't worry about memorizing. Just think. Think. What's happening in the body? That's right. Think. I'm doing everything I can to try to get you to think and not be like those other medical students who are going through the motions. Oh, what am I supposed to do? Go into this hole? I'll walk into the hole. Why? Because first aid says. Or can you think of the answer? There it is. Just think. All right, now what's coming up here is, now we're done with carbohydrates. Now we're going to finish off this hour starting off with lipids on chapter 15. So we'll do lipid synthesis here and storage. Now this chapter has a lot to do with fatty acid issues, fatty acid synthesis, and everything in this chapter about cholesterol is in this chapter as well. So a lot of cholesterol issues in this chapter. And then tomorrow we'll do, so we'll finish off cholesterol tomorrow. We won't get it to it today. But the idea is we're going to do chapter 15 today and tomorrow in the first hour or two. Then after that, we'll get into fatty acid oxidation tomorrow. So the rest of this hour, as well as all tomorrow, we're going to do lipid stuff. And then on Friday, we'll finish off doing amino acid and bilirubin and iron metabolism on Saturday. And purine stuff, purine salvage, and so on. Major day is Friday. No lecture on Saturday. Somebody asked that earlier. What about Saturday? No lectures on Saturday. Okay, Rouge can't control laughing. He acts so funny. You gotta have a sense of humor, right? You gotta have a sense of humor. There's a lot of content, but you gotta sprinkle it with some humor along the line here. Okay. All right. Now, what's coming up next is the issue of fatty acid synthesis. Now, let me say something about fatty acid synthesis on page 219. There was two separate medical students over the last couple of years that told me they had two questions on this page, two questions about fatty acid synthesis. So I'll tell you what I heard once we get to those parts on this particular page. Okay, so the idea is when do you make a fatty acid? Everybody knows that you make fatty acids by eating a high carbohydrate meal. You eat a donut, you're going to make a fatty acid out of that. Now, to make a fatty acid, just like making a cake, you need ingredients. And the ingredients to make a fatty acid is you need large amounts of acetyl-CoA, large amounts of NADPH, large amounts of CO2, and large amounts of ATP. And where is the pathway of fatty acid synthesis? It's in the cytoplasm of most cells. So most cells, including brain cells, brain can make fatty acids, but again, in the cytoplasm. The brain doesn't make much fatty acids, but it can make fatty acids. Here. Okay, so what we to orient ourselves is on the left side is the mitochondria and on the right side is the cytoplasm. Now, if you were asked, where is all these things coming from? The ingredients to make a fatty acid. For example, if you were asked, where's all that CO2 coming from? Used to make a fatty acid. And you would say, oh, come on, man, that's an easy question. CO2 is dissolved everywhere. Ask me something harder. Okay. Where's all this ATP coming from? Used to make a fatty acid. And you'd say, come on, man, come on, you're insulting my intelligence. You eat a high carbohydrate meal, you run glycolysis, making ATP, down to pyruvate. Pyruvate goes in the mitochondria, sees PDH, generate ATP there, generate an acetyl-CoA, condenses with OAA in the Krebs cycle, which we purposely skipped today. And then it goes in the Krebs cycle, making more ATP there. Ask me something harder. Okay. Where's all that acetyl-CoA coming from? Used to make fatty acids. Oh, you say, come on, here is the acetyl-CoA. Wrong. Notice this is acetyl-CoA in the mitochondria. Fatty acids are made in the cytoplasm, and there is no transporter for acetyl-CoA from the mitochondria into the cytoplasm. So here it is. If they ask you, what's the source of the direct source of the acetyl-CoA? You answer by saying, by eating a high carbohydrate meal, you're going to run the Krebs cycle, working, cranking out the ATP. And by doing so, you're eventually going to shut down the top enzyme of the Krebs cycle, which we purposely skipped. And this enzyme is called the isocitrate dehydrogenase, and by inhibiting that top regulated enzyme of the Krebs cycle, then citrate will accumulate. Citrate will shuttle out of the mitochondria into the cytoplasm, and a low yield enzyme, not even shown here, that cleaves the 6 carbons of citrate to the 2 carbon acetyl-CoA, and that is, talking back off there, the acetyl-CoA is now the source of the carbon to make a fatty acid. And then the other product is OAA. So if they ask you, what is the direct source of the acetyl-CoA to make a fatty acid in the cytoplasm? You say citrate. Now, indirectly, it was glucose, that's in the high carb meal, but directly it was citrate, in case they ask you. Now, why am I going through the motions here about where these things are coming from? Because according to one of the students, in my opinion, this is a low blow, but according to one of the students, they asked, where is the NADPH coming from used to make fatty acids? So I'm trying to cover all the bases in case they ask you about the acetyl-CoA. But according to the students, they ask him, where is the NADPH coming from? Now, unfortunately for him, he got it wrong, because he was looking for the option, the hexose monophosphate shunt, which would have been correct, but it was not an option. What they were looking for is this, is the hexose monophosphate shunt is one source of NADPH. But there's a second source, and it is this oxaloacetate. Nothing goes to waste. It forms malate, no big deal here, but then there's an enzyme that was on the boards called malic enzyme that cleaves malate to pyruvate. That's not a big deal, but importantly, it generates additional NADPH. So one more time, there are two sources to generate NADPH to make fatty acids. The hexose monophosphate shunt, the logical dominant source, and the other is malic enzyme. Kind of a weird name of an enzyme, but that's the name of the enzyme, malic enzyme, on the national board. In my opinion, low blow. But what can I say? Hey, don't go blaming me. Don't go. Hey, remember, I'm just the messenger. I know a lot of you out there are blaming at me, and you're probably throwing things at the computer screen and my picture over here. Remember, I'm just the messenger, according to the student here. Don't go blaming me. I know that's picky. I know. Hey, Takir, they're not your friends. The board people asking those questions are not your friends. Never assume they're your friends. So what do you want me to, it's a low blow. All right, that's a lot of low blows. That's right. But what can you say? They're not your friends. Okay, so now we have accounted for all the goodies used to make a fatty acid. And now we're ready to make the fatty acid. And now we come to the dominant enzyme of fatty acid synthesis. And this was the second and last item that I heard about being on boards regarding fatty acid synthesis. This is the top enzyme, the top gun of fatty acid synthesis. And this enzyme is called acetyl-CoA carboxylase. Now, according to a student, they were asking about the regulation of this enzyme. Now, don't worry about the regulation now. We'll talk about it in a minute. What does the enzyme do? It tells you. It's called acetyl-CoA carboxylase. And so what the enzyme does, it uses CO2 to put it onto acetyl-CoA to make the 3-carbon malonyl-CoA as a product. And to do that, it needs CO2, biotin, and one more thing. What's the other thing this enzyme needs to work? It needs biotin, CO2, and one more thing, ATP. Now, how'd you know it needs ATP? Yes, that's correct. How'd you know it needs ATP? That's right. Well done. People are thinking. Because this is now the second of the 3 ABC carboxylases. All 3 of them are critical. This is the second of the 3. I know what you're thinking. What was the first of the 3 that we talked about earlier? What was the first of the 3 ABC carboxylases? That's right. That was pyruvate carboxylase on the left side of the figure here. Pyruvate carboxylase in the pathway gluconeogenesis. And tomorrow we'll talk about the third example. So this is the second of the 3 ABC carboxylases. All right, now once it makes this malonyl-CoA, we'll come back to the regulation of the enzyme in just a few minutes here. Now, once malonyl-CoA is formed, it's a precursor. And then we come to the enzyme that actually makes the fatty acid, called fatty acid synthase. Now, depending on where you went to medical school, your instructor in medical school may have spent a whole hour of your life talking about the mechanism of fatty acid synthase. Forget about it for the boards. Useless information. Don't worry about the mechanism of the enzyme, fatty acid synthase. All you need to know is this enzyme, fatty acid synthase, is taking multiple malonyl-CoAs, takes 2 of the 3 carbons, condenses them together, and so the fatty acid is growing 2 carbons at a time. 2, 4, 6, 8, 10, 12, 14, 16 carbons at the very end. And when it builds the fatty acid, it's then using the electrons from NADPH to put the electrons back into carbon. At the very end, the mechanism, forget about it. At the very end of all the synthesis, the enzyme spits out palmitic acid as the product. 16 carbons with no double bonds. It's a saturated fatty acid. It is the only fatty acid that human beings can make. So on board exams, you might see this enzyme called fatty acid synthase, or you might see it called palmitate synthase, because it's one and the same. You only make palmitic acid as the product here. Only fatty acid that human beings can make de novo, from scratch. And you're going to say, hey, I thought human beings can make other fatty acids. That's true, but not from scratch. What happens is the fatty acid can then go to the ER, endoplasmic reticulum, where elongation can take place. You can elongate the fatty acid by enzymes you don't have to know, to C18, C20 fatty acids, and also desaturation enzymes, desaturases in the ER can put double bonds into the fatty acid to make unsaturated fatty acids. The key point here is that you need to know, in humans, there is no desaturase enzyme that can put a double bond beyond carbon 9. In other words, we have desaturase enzymes in the ER that can put a double bond at carbon 5, carbon 7, carbon 4, carbon 9, but nothing beyond carbon 9. Let me ask you this. What is the significance of that yellow box statement? You must know this as a physician. What is the significance of this statement that humans have no desaturase that can put a double bond beyond carbon 9? What's the significance of the statement? That's just words, but what's the significance of it? Not irreversible. Good try. Good try, Lily. And trans, no. Daniel, what about? Now we're getting somewhere. And this is why 2 fatty acids are essential. There are 2 essential fatty acids. In other words, you must get them from the diet. Does anybody know? They both begin with the letter L. What are the 2 essential fatty acids? That's right, linoleic acid. Look at the nation, so on. Linoleic acid, and the other is linolenic acid. That's right, Daniel. That's right. So you got both of them there. Fumni, that's right. Tiffy, that's right. So the idea is there are 2 fatty acids that are essential: linoleic and linolenic acid. And why are they essential? Because linoleic acid has 2 double bonds. I doubt you have to know the exact detail, but it has 2 double bonds at carbon 9 and 12. We could do the one at 9, but not the one at carbon 12. Linolenic acid has 3 double bonds at carbon 9, 12, and 15. We can do the one at 9, but not the one at carbon 12, and not the one at carbon 15. And so that's why they are essential. And as a result, then when you eat linoleic, linolenic acid, you can make arachidonic acid And in pharmacology, you know arachidonic acid can be used to make the prostaglandins and so on. So you must know this yellow box statement. Why 2 of the fatty acids are essential? Because we cannot put double bonds of carbon 12 and 15. And you need those 2 to make the prostaglandins. High yield stuff you'll talk about in pharmacology. But this is the biochemical basis here. All right, now in endocrinology, this enzyme here, acetyl-CoA carboxylase, as I mentioned, it is highly regulated enzyme, and that was on the boards. As you would predict, insulin turns on the enzyme. As you would predict, glucagon turns off the enzyme. No need to get into the detail. You know how they work, these hormones. You must know how they work. They work in by phosphorylation, dephosphorylation mechanisms, and so on. And insulin also turns on the gene for the enzyme. Remember, insulin can turn on gene expression. And by the way, and also citrate allosterically turns on the enzyme. So what I'm getting at, this enzyme, acetyl-CoA carboxylase, is regulated all 3 ways. It's regulated at the gene level by insulin, regulated by phosphorylation, dephosphorylation level by insulin and glucagon, and allosterically by citrate turns on the enzyme. Now, one of those items about the regulation was on the national board. I don't know which one, but one more time. If they ask you, how do you regulate acetyl-CoA carboxylase? It's regulated at the gene level by insulin, phosphorylation, dephosphorylation by insulin and glucagon, and allosterically by citrate. This is why, now bigger picture, this is why most of the fatty acids in the human body are made by the liver. Other cells can make fatty acids, but 99.9% of the fatty acids are made by the liver. And you're looking at the reason why. Remember earlier, when you eat a high carbohydrate meal, all the excess glucose funnels to the liver by the GLUT2 transporter. Once glucose enters the liver, insulin takes command of the liver and tells the liver, turn on glucokinase, turn on PFK2 to turn on PFK1, drive glycolysis down the pyruvate. And while all that's going on in glycolysis, insulin comes over here and turns on acetyl-CoA carboxylase. So the enzymes are ready for fatty acid synthesis. How beautiful this works. You can memorize all you want, but notice the beauty. What insulin is doing in the liver is taking command here. All right, now these fatty acids that are being made in abundance in the liver, do not stay in the liver. What happens is they get packaged in triglycerides, reforming into triglycerides. Now the biochemistry of that is not important. What is important is the triglycerides are then packaged in the liver in a particle called VLDL that we'll talk about tomorrow in much greater detail. And VLDL is a huge particle that's now composed of mostly triglycerides, now leave the liver, made by the liver only, VLDL, and ships it into the bloodstream and picked up by the adipocytes. And in the adipocytes, as we'll talk about tomorrow, the triglycerides are delivered here in the adipocytes here. All right, now one more item about this figure. Major item. And that is in alcoholics. Now let me ask you this. Every medical student knows this. What does the liver look like in a long-term alcoholic? Let me see your answer here. What does the liver look like in a long-term alcoholic? That's right, it looks fatty, the fatty liver characteristic of an alcoholic liver. All right, but now why? You can memorize it as they have a fatty liver, but why is the liver fatty? And now you could say, well, it's because alcohol is highly caloric. It is highly caloric. Yes, you make a lot of triglycerides. Yes, yes, yes. But that's not the problem. That's not causing the fatty liver. What alcohol is doing is this. Remember, if alcohol is making triglycerides, VLDL will ship them out. They would not accumulate in the liver. The problem with alcohol is just put a big X on VLDL. It inhibits VLDL assembly. So triglycerides are being made from the alcohol because it's highly caloric, but can't get out to the liver because alcohol inhibits VLDL assembly. Now here is the pathology. On the left side is a normal liver, rustic color. On the right side is a fatty liver, that lighter color. And if we were to do a histological examination, then here are the hepatocytes. On the left side are the normal hepatocytes. On the right side are the hepatocytes from a fatty liver. And notice the lipid droplets. Triglycerides are accumulating in the liver. Remember, triglycerides cannot go through a membrane. To get out to the liver, they have to be packaged in VLDL. And if you inhibit VLDL assembly, the triglycerides stay behind in the liver and get these lipid droplets that go on. And by the way, how long does it take for your liver to look like this? Something like 2 or 3 weeks of binge drinking. Your liver will look like this. It doesn't take long. It does not take long for your liver to look like this. But fortunately, it's reversible at this stage. But as time goes on in an alcoholic, long-term alcoholic, then what happens is because of so much triglycerides in the liver, the liver becomes cirrhotic, and then the liver starts to scar. Let me ask you this. In histology, what is the name of the cell type in the liver that scars the liver? What's the name of the cell type that scars the liver? What's the name of that cell type that scars the liver? Not the Kupffer cells, good try, no, not the stellate cells. It's called, not the fibroblasts. And the answer is, it is the Ito cells, ITO. The Ito cells are the cells responsible for scarring of the liver. That's right. There we go, Natalie, Angela. That's right. It is the Ito cells. Whether you call them Ito or Ito, it's one and the same thing. But that's the, so when the Ito cells start to scar the liver, it's irreversible, damaged liver, major item. Now, one last figure, won't just take a couple minutes. There's one last figure here, then we're done. And that is, what is a triglyceride? Because you make the fatty acid, so now what makes sense? What is a triglyceride in the first place? And what a triglyceride is, it's where the hydroxy group of glycerol and the carboxyl group of a fatty acid, they condense together to form an ester linkage. And that is an ester linkage. Let me ask you this. You know hydroxyl groups are polar. You know carboxyl groups are polar. What can you tell me about an ester linkage? This is the ester linkage. What is an ester linkage, polar or non-polar? That's right, it is non-polar. And so the idea is that you take the 3 fatty acids that are being made and you stick it in ester linkage onto glycerol, making a triglyceride. Why do you make the ester linkage? Because triglycerides are 100% grease. There's nothing polar about a triglyceride. It's 100% grease. No water accompanies a triglyceride, completely anhydrous. That's why you make a triglyceride, because no water accompanies them. Now the enzymes which make the triglyceride are not important, but the enzyme which cleaves the ester bonds of the triglycerides are called lipases, and there are many lipases we'll talk about tomorrow. For example, lipoprotein lipase. For example, hormone-sensitive lipase. And in physiology, you talk about pancreatic lipase. But by definition, lipases are enzymes which cleave the ester linkages of triglycerides. Now, where else do you have ester linkages? You have ester linkages in phospholipids on the right side. Here are two fatty acids in ester linkage to glycerol, and on carbon 3, in the phospholipids, there's a head group, like inositol or choline or glycerol. And the class of enzymes which cleave the ester linkages of phospholipids are not called lipases. They're called phospholipases, and there are many phospholipases. For example, there is an enzyme called phospholipase A1, and phospholipase A1 cleaves the fatty acid off of carbon 1. In pharmacology, you talk about a phospholipase there called phospholipase A2. You also talk about it in hematology. And in phospholipase A2 cleaves this bond right there, liberating arachidonic acid. And you know, in pharmacology, arachidonic acid is used to make prostaglandins. And in hematology, phospholipase A2 cleaves off arachidonic acid used to make thromboxane A2 that does the vasoconstriction that goes on. So again, these phospholipases here. Let me ask you this. Was there any other phospholipase that we talked about in biochemistry? Any other one that we talked about? Yeah, that's right. And that's phospholipase C. Let me ask you this. What receptor was turned on to ultimately turn on phospholipase C? What receptor, without looking in the book? Don't say GQ. GQ is never, ever, 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 never, never, ever, ever a receptor. What receptor was? Don't worry. I'm being dramatic here on purpose. That's right. It was alpha 1. So when epinephrine binds the alpha 1 adrenergic receptor, in turn, it turns on GQ. Alpha Q binds GTP, which turns on phospholipase C. And what was the lipid that was cleaved by phospholipase C? What was the lipid again that was cleaved by phospholipase C? PIP2. And what is PIP2? You're looking at it on page 221. This is PIP2. And what does phospholipase C do? It cleaves this bond right here. And by cleaving that bond, what were the two fragments that were generated by phospholipase C cleavage of PIP2? What were the fragments again? Diacylglycerol. And what's diacylglycerol? You're looking at it in the orange. Diacyl, two fatty acids, diacylglycerol. And what was the other fragment? IP3. And what's IP3? There it is in the purple outline. By cleaving that bond, it generates the two fragments, diacylglycerol and IP3. There it is, the second messengers. Okay, everybody, we're done for the day. So I'd like to thank Carrie for wonderful help she had with the technical issues, and Dr. Aaron and Dr. Seda for the Q&A help here. And so we're done for the day. So tomorrow, again, we're going to do lipid stuff, a lot of lipid stuff, a lot of cholesterol. If you want to know what LDL is all about, HDLs, why is it good and why is it bad, stay tuned tomorrow. A lot of farm, a lot of path tomorrow. All that stuff of type 2A, hyperlipidemia, major stuff for your career as well as the boards. And then after that on Friday, the other issues of chapter 18 and 19 on bilirubin and stuff. So again, major stuff coming up over the last 2 days. And please come back over the last 2 days. I'll make it worth your while. Okay, everybody, we're done for the day. Thank you so much for attending and for participating. Thank you so much for your comments on the public chat pod. I appreciate your wonderful comments. It means a lot to me when you make those really, really nice comments. It's part of my reward reward to see the great comments that you make. Thank you so much. Okay, everybody, so we're done for the day, and I will see you back tomorrow.